there's quite a bit of research now that shows that women in general are disproportionately impacted severely because of COVID, not from just exposure to COVID. It's because of the impacts of COVID. They'll have to stay home and watch the kids because the kids' schools are closed. And so they end up actually getting just across the board. I think maybe this is my soapbox, but just being a woman anywhere in the world is difficult, just because of any number of challenges in the society. In a pandemic, it's even worse. They're just exposed to so many different challenges of taking care of the family, taking care of the job, taking care of everybody else. I think that's the impact that has been demonstrated.
Thank you all the dignitaries for taking all the queries. Now I would like to request our esteemed chairperson of today to kindly felicitate our resource speakers, guest speakers with memento and souvenirs. I'd like to call upon the dais Dr. Payal Thavde ma'am, head of the department, postgraduate teaching department of law, for formal vote of thanks for this session.
A very good noon to all of you. Indeed, it was a very, very thought-provoking session, and the title itself says that COVID-19 pandemic, a review of disaster management and lessons for future. And I think all of you will agree with me that we all are connected with this topic, irrespective of the thought that whether we know science or not. So, on behalf of 108 Science Congress, Indian Science Congress and Rashtrasant Tukadoji Maharaj Nagpur University, I express my heartfelt thanks to our today's chairman, Dr. Manmohan Parida sir. Sir, you have said that you have been here accidentally, but I think, and all my participants will agree with this, you are not accidentally here, but you are destined to be here because of the topic. And ladies and gentlemen, please give him a huge round of applause, because I being the coordinator for this session, I think it is my responsibility to introduce you all that why I am saying this. Sir, being a virologist, he has core areas of research which encompasses strategic biodefense research and development, with significant contributions in the field of rapid detection technologies, antiviral drug development, new generation vaccine candidates, molecular epidemiology, genotyping, phylogenetic evolution, and trafficking of emergence of viruses of biomedical importance. Sir has closely worked on COVID-19 related aspect, particularly the RT-PCR and RT-LAMP technologies for large number of viruses, and he also worked on dengue, chikungunya, West Nile, Nipah virus, flu, SARS, COVID-19 and other. He has the distinction introducing the LAMP technology in India. He has also developed RT-LAMP tester for high-risk biothreat viral agents via Ebola, smallpox, CCHF, AFD, Nipah virus, etc. The swine flu RT-LAMP technology, he has been transferred to Messrs RAS Life Sciences and has been approved by ICMR with DCGI clearance as an alternate indigenous technology to WHO approved CDC RT-PCR. He has also established the test and evaluation facility, synthetic blood penetration, etc. for COVID-19. He was the member of various interministerial task forces who have closely worked on such issues wherein so many threats like COVID-19 has been there. And if I go on, I think I need another half an hour to read his biodata. And certainly being a law person, I am not well versed in all these super huge medical technologies. So thank you so much, sir, for joining us here as a chairperson. At the same time, I express my respectful thanks to Dr. Kumar Ralapalli, sir, for joining us all the way from Jacksonville and sharing with us your expertise in this area. I also extend my heartfelt thanks to Dr. Anand Srinivasan, sir, who have joined us from Singapore. So thank you so much, sir, for sharing your views and expertise on the area of COVID-19. So I also express my heartfelt thanks to my vigilant participants who are throwing some really thought-provoking questions to the panel. This is what we are here for in the Indian Science Congress. So thank you so much, all the participant delegates and the chairperson. Thank you so much. Ladies and gentlemen, we'll be beginning with the next session very soon within a break of 5 minutes. So you all can go and please come back. We have another thought-provoking session coming to us. Thank you so much. Thank you.
Chaube, Assistant Professor from Postgraduate Teaching Department of Law, Rashtrasant Tukadoji Maharaj Nagpur University, extend a very warm welcome to all of you on behalf of 108th Indian Science Congress and Rashtrasant Tukadoji Maharaj Nagpur University to the plenary session number 10 on Project Management Theory and Practice in Academia, Biotech and Pharma Industries. Before we proceed with the session, I would like to give few instructions to the participants. Kindly keep your cell phones on a silent mode. Please be seated throughout the session. In the case you have any queries regarding the session, it will be taken at the end of session with the permission of the Honorable Chairperson. For this session, it is a matter of pride and privilege to have Honorable Dr. Rohini Dhulipala, Director CMC, as a chairperson. I request Honorable Chairperson to kindly raise the hand. Welcome, ma'am. I would like to introduce to you our today's esteemed chairperson, Dr. Rohini Dhulipala Madam. Dr. Rohini Dhulipala Madam completed her master degree from SV University, India and worked as a research specialist at Jefferson Medical College and University, Pennsylvania, Philadelphia, USA. Dr. Rohini is instrumental in developing fluorescent-based conformation-sensitive gel electrophoresis CSGE for detecting in 6 unique mutations in BRCA1 and adapting this method to conduct genetic diagnostics at the Penn Medical Center. This work led to few publications in very reputed journals like journal Clean, INV, Human Genetics, PNAS, etc. Dr. Rohini Madam transitioned from academic research to biotechnology industry and worked as R&D scientist. Madam also worked as manager for manufacturing division producing various genomic reagents at GE Healthcare. As a scientist at Amicus Therapeutics, she contributed to the identification and use of small molecule drugs for treatment of rare and orphan lysosomal disorders like Pompe and Gaucher, and her work led to develop several drugs like miglustat. Dr. Rohini got master certificate in project management from Villanova University, Philadelphia. She served as a project manager and program manager at Life Technology, Vetana, Roche and Lonza as program manager and led several programs starting from early phase process development, phase 1 2 and 3 of clinical phase and commercial program for small to big pharma biotech clients. Dr. Rohini got 5 patent applications from her industrial job findings. Welcome back. Now I would request the chairperson, Dr. Rohini ma'am, to kindly proceed with this session. Also, I would like to request Dr. Payatore ma'am to kindly welcome our today's chairperson with memento as a token of appreciation and gratitude.
General talk on there are young people in this hall, and then I heard there is another room full with young people, so I wanted to give them an opportunity to give a project management as an opportunity and a career path to people to take. Just to give you a background of myself, don't know what happened. Maybe I can touch the podium. So just to give you, I started in academics, worked as a lab scientist in various roles, and slowly I grew up in my career, and I found program management as my passion and pursued that as my path of career. So just to give you the overall, I'm going to take you up to 30,000 view and all the way to the little more details of 10,000 view of program management. There is some basic fundamentals as well as how that can be used in pharma biotech as well as other industries in order to produce the product from conception to commercialization. What are all the principles and tools that we can use and the deliverables that we need to accomplish in order to meet that project or program or portfolio level achievements. Okay? So my agenda today will be covering mostly the project management, few basic slides with the fundamentals as well as the skills that require for a good project manager or a program manager, and then the project management of various steps that you can take through the biopharma industry, and then a quick summary. What is program management? I think it's the process that is going to drive the value in an increment way to achieve the program deliverables, despite of the emphasis of artifacts and deliverables that we wanted to reach. And there are multiple things, the strategies and the planning that is involved. At the end of the day, a project will have a duration that you will be reaching, the start to finish. The program will have multiple projects involved, and furthermore, if you go into the portfolio management, it is business critical that will be deciding and then further moving into that. So a program and the project are the critical endeavors in any business to pursue and to get the product out in a way. Once the academia finds some new research that is useful for the humans, that can be transmitted into a commercial scale that needs to go through any industry. As a matter of fact, it can take a IT industry, software, anything. It goes through certain processes, and that is where the program management will come and help that. And if you take this, analogy of a train, a project is like a train that is going to a station. But if you take a program, that is like a central station that various trains will be coming in, and then you synchronize it and you coordinate those efforts in order to meet that objective or the goal. So here, just wanted to emphasize various layers of project management. If you can see the overall portfolio management at a very high level, the program management is like a business level interest that a company or some kind of an organization that will have, and then that will drive into the program management, where you'll have the stakeholders involved and their interest to drive, and then further down into a very quality-driven, deliverable-driven project management, where you have the real work to be done, and then you submit or you release that, you go to the commercial launch of that product. So you have a different stakeholders, different requirements as you progress through from project to program to portfolio level. This is a very example that I wanted to put together. This is from my current work that we have been working on. As you can see, at a project level right now, we worked on the COVID project. I'm glad to see Mr. Anand provided one of our vaccines from our company, Novaxovid, which was launched early this year. So in order to develop that and further what is being done for the COVID-19 project, there are multiple projects that are going on. How that product is being presented, whether it's a 10-dose vial or single-dose vial or pre-filled syringe format, or what can we improve in order to do that. Those are all comes under the individual projects, the multiple projects and the small ones. They will roll up under the COVID program, where you have a business need and the commercial will team say, hey, for Australia, I need only pre-filled syringe. Hey, for US market, I need 5-dose vial or unit dose vial. So those are all the things that will be worked at the program level, and all these individual programs will feed into the program and further up into the portfolio. The business at the corporate level will decide what is the strategy at the topmost level, and further comes down in a way that it can be disseminated and worked and then produced back and rolled up into the management level and further to the provide the business opportunities to the company. So there are a few models of project management methods that are currently being available. The typical water flow model, and then Agile, I'm sure you all must have been heard, and then the Scrum, and then Kanban. You all must be various forms. If you're in IT, you hear a lot about Agile Scrum, and then Kanban, but if you're in the pharma biotech, you will see very typical waterfall model and Kanban. And if it's a process improvement projects or programs, you will see Kanban as well, okay? And then the types of program managers across the industries, you can see pharma biotech, healthcare, IT, software development, energy, as well as construction. This is just a handful. But if you see typically, even if you're in academics, if you're running a study or a project, that itself is a small project to you. You can implement certain tools and work through those. That's how you grow and make it much more. Having all organized and then providing that information when you come to the publishing the paper, you'll have everything organized in a way that can be put together in a more organized way. So program managers, what do they really do and how they interact is they are the bridge or the central location. In some certain industries, they'll say they are the mini CEOs because they have the information that comes from the various sources. They build the bridge between the operations, business, and the customers or internal sponsorship requirements. Furthermore, providing it into the regulatory agency and get the approvals, whether it's a pharma biotech or whatever the industry you are in. They connect the information, they bring the stakeholders. I have a couple of other slides coming up. I'll show you, but they are the bridge between the various functions within the organization or across the organizations externally. So customer-centric, so you can see the program manager is like in the tower, handling the information that comes from various cross-functional teams that will be working, whether it's a project manager, he is a tactically driving all the information to achieve the project objective, and each individual group have their own deliverables or requirements that they will be working towards the main project. And the information will come to the center, to the project management, and they will make sure that they are meeting the right deliverables on time and then within the timelines. And if there is something that goes off track, they will escalate to the appropriate stakeholders, internal or external. So they are the central liaisons between the cross-functional teams and making sure that scope of that project is fully achieved. So it's like a throughout the life cycle management, from all the way from the scope they will drive and all the way to the closure of the project. So what does really a project or program manager do? There are 5 fundamental principles the project management will be driving. That is from the initiation of the process, where there is a feasibility or a basic idea, and then the planning. You all might be heard that 30% of the project management planning is very critical. Without planning, I don't think you'll be successful. For any success of the program, you need to have a fundamental planning. So the planning part of the process is critical. Once it's planned, a charter and a stakeholder element is completed, then starts the execution, where that scope is translated into multiple disseminated tasks where the cross-functional teams will achieve and provide those deliverables. And then the monitoring and controlling during the timelines where each one has to be completed. I'll give you the practical example. That's where the actual work will be coming, or the monitoring of the work of the PM will be doing. And then the project closure. Once a project achieves its objective, it will be closed, completing all the audits and whatever it's needed depending on the project requirement. So every beginner possesses a great potential to begin with an expert is our own chosen field. Just like that, a project manager has a multiple layers of traits and the qualities that is needed. When you start your career, you'll have few things, but as you go through the process and learn, there are multiple layers of complexities that has to be worked out in order to progress through the ladder of the program management, from project to program to portfolio level. Some of the essential skills as I listed here, execution, making sure that scope is fully vented, it's understandable and it is within the expectations of the company objectives, that scope has been driven and executed. And then the strategy development, in order to achieve that objective, what are the strategies that is needed? If it's take a biotech pharma product, API, or like a vaccine, what is the regulatory strategy? What is the commercial strategy? What is the analytical method strategy? Where do we manufacture this product? These are all some of the critical strategies that one has to work, and this is driven by cross-functional teams. It's just not the program managers or project managers, but they facilitate and bring those questions asked in the right way and put together and document it so that we can, when it comes to the filing or transferring the information, you'll have all that information. And the critical leadership, of course, you have to work with your team and then encourage them, empower them, and because these team members, they are not reporting to the project managers, they are indirect, so you need to have that skill to influence them and work with them. That leadership skill is very important. So the empathy is the fundamental thing for the success of the project manager. Along with that, I see a lot of lists here, but I'll cover a few of them, but these are agility again. You have seen, everybody has seen during the COVID, there's a constant change in, the decisions and how fast things has to make. 7 years or 10 years of a protein vaccine to be made, it has been cut down to like 16 or 2 years process. That change, a risk-based approach of the organization will have a lot of changes, and one has to be very fast in accommodating and making sure that all those changes have been captured so that the information is, some question comes up, why certain decisions has been taken. Conflict management, yes, it is one of the critical things. As you are working with multiple teams, you will now not have a direct report, so you need to understand where they're coming from because they have their own priorities. Getting the work done without a direct reporting is a critical factor for project manager or program managers. Negotiation, because the timelines are critical for every program manager, their project is important, so those are all the things that comes like going to the leadership team and the functional heads and asking for the additional resources or prioritization. All these things comes under the negotiation and the decision making as well. The part of the decision making is like, the critical strategies that are taken and getting that alignment from top down and bottom up is very important in order to be a successfully achieve that project goal. Technical competency, sometimes you'll hear that project managers don't need to have any technical information if you follow the tools and the techniques, but if you have the technical background, it helps you to understand your conversation and how to drive the project for success. It's not needed in certain industries, but it's good to have and try to show that it will only take you to the higher levels faster than what you can do without that limitation. So don't limit yourself, try to get into the weeds and try to understand so that you can ask the right questions. And then business acumen. So it's just just not your project, but you also have to roll up to the business needs. So that is what important. If you just do your project, hey, I'm done, that doesn't help you. You need to connect with your project, why you are doing this, and that will make you help your team to achieve the goal faster. So it is very important that you understand the business needs and translate that and inform your team in order to achieve the success of your project. Influence, of course, you need to have that influencing skills. And again, all these are critical in order to be, this is the industrial standard, anyone, but I don't think anybody who started their profession will have all these skills. So as you progress through, you will evolve and learn these skills, and that's where your passion and your drive for, how you can improve your projects from project to the portfolio level and multiple projects to the PMO level you can grow up to. Some of the soft skills as a project manager, you should be able to work with multiple cross-functional teams in order to present the information to the leadership, the executive VPs, CEOs. You need to be able to prepare this and provide in a way that if it's a technical, it has to be technical, and it has to be a leadership level. You don't need, there's a certain way of filtering the information, but still presenting the facts is the critical thing. So that's where your communication skills as well as the presentation skills are very critical for a project manager, both verbal and written communications. These are all the critical things because you yourself is not an expert on anything, but you will be getting the information from experts and communicating that, the status updates to the leadership, and that's where that articulation of the information is very important. And again, people skills, managing without the authority is the fundamentally a critical factor for all the PMs to drive for the success. Some of the traits for the success also is like intelligence and the creativity, how to present the information to the teams or the people who really want to understand what it is the question at. Is sometimes people keeps talking on and on, but it may not give the point that you wanted to convey. That is where the project manager will come and ask, because technical people have their own way of conveying the message, but not everybody can understand that. So it has to be a balanced way of presenting the information to the right stakeholders in the right way. Again, you have to be problem solving and then self-motivated in order to drive, because everybody have their own interest. You are the one who is holding the torch and trying to get this project done, so that's where your self-motivation will come in. So some of the helpful resources that I've been using and people can use is like Toastmasters, where you can develop your presentation skills as well as your leadership skills, and then Project Management Institute, you can get the PMP certification from there, and there's a lot of trainings and there's a lot of information on how to run the projects of various types is available there as well. And the employee will have their own trainings. Each employee or the companies, they have their own training that they are catered for that specific. If it's a pharma biotech, they will have that kind of a focused training for project management. And then universities do offer, I did mine in Villanova University, but every university nowadays have the program management course. And then LinkedIn, you'll have multiple trainings on the LinkedIn as well on various aspects of program management. So now I wanted to drive to a real example of my current work and what I'm doing right now. Again, CMC stands for Chemistry, Manufacturing and Controls. This is the fundamental first step that's where the pharma biotech work will start. If there is a product or a vaccine or an API that is a molecule that they wanted to establish, critical things to establish is like early phase feasibility, right? The portfolio entry. Once the research and development people identify something and they found that it is a lead candidate after the feasibility, then it enters to the CMC portfolio entry. That's where the first example. At each of these life cycle management of these steps, you will have like a milestone review. At each of this review, you will present what is being completed, what you will be completing or coming up next to the stakeholders, so that you are clear on your expectations of your project, what has been completed. So at the entry of the project, you will have a different requirements. You just need to show at a lab level in an R&D environment, it shows the gel, it works at a very early stage. But later on, when you progress through the life cycle, when you are at a clinical phase or preclinical phase, you have to show in the tox batch whether it shows feasibility in animal model or not. Then it goes into the pilot batches and like clinical trials, and then into the tech transfer, and then the commercial production, and then the commercial product launch. Each one have their own requirements, which I will be walking through at a high level again, the requirements of each of the phases that as any pharma biotech company will be following. This is the waterfall model that I will be following. This is a standard FDA for any design input, design output, and the verification, validation. All these requirements are very standard waterfall model that is followed in most of the pharma biotech, but there are other models that is followed as well. With this example, I'm going to show you some of the requirements at the early stage, what is needed in order to start a project, a business case, what is the business need for this project to start, and then resource, budget, timelines, how long does it take, overall that is critical. And depending on the project, whether it's a API, and depending on the industry as well, if it's like a small company, they don't have all the manufacturing resources, they will be relying on the external manufacturing. So which CMO I should pick, which development partnership I have to make, all these things will be starting at the very early stages. And as you progress through, you will have like a next step, what cell line I should use, what is the best one that is, and what is the tox batch, where do I produce my drug substance, and what is the testings I need to do, and what is the shelf life, what is the standard, reference standard I have to use. These are all the critical, I'm going into very much details right now, but these are all the different deliverables that during the development of a vaccine or a product, API, that these will be used as the tools or the checklist for people to accomplish their project goals. Same thing again, if you're going to, phase 2, phase 3, you'll have certain different types of deliverables that you will be working towards. And again, as the complexity will increase, when you go into the combination medical device, will have an additional requirements which you will be working with the cross-functional teams in order to get the alignment. Similarly, when you're close to the commercial, you'll have a validation package and you have to submit to the regulatory agencies. All that package has to be worked through, the program management will oversee working with cross-functional teams, with the experts and SMEs to build that package and then produce to the agency to get the approval, whether it's emergency use authorization or whether it's the, pre-market authorization or post-market. All this has to be mapped out, and then timelines will be established, and the project manager or the program manager will be driving those activities. So that brings me back to the summary slide. So as a project manager or a program manager, to be successful, you need to have a soft skills as well as some of the analytical critical skills, and you have seen the differences of project, program, and the portfolio differences, and the trains that is needed, and also the waterfall model of detailed what are the deliverables that is required in order to be successful to transfer an early stage to all the way to the commercial, and then the soft skills is very critical for the success of the project manager. Any questions? Yeah, okay. Oh, okay, I see. I think towards the end. Okay. Yeah, sure. Okay, thank you. I think we'll hold the questions to the end. Let me close this one, and I wanted to all right, I think I will introduce my next guest. So, thank you. you everyone for participating in my talk, and I will take it to the next presenter, which is Satyapi Kunapalli. He is the director and the Soul Sherry Thrombosis Research Center. He received his PhD from the IISc Bangalore. He started his career, first position in the University of Texas Medical Center in Galveston, Texas. In 1988, he moved to Temple University, started his career, and now he is a professor in cardiovascular sciences. And his main work, he has worked over 20 years, and he has published over 200 papers and contributed towards the platelet physiology and signaling. I invite now Dr. Kunapalli to the stage to present his talk.
Nice introduction, and I thank the organizers of the Research Congress for inviting me and giving me an opportunity to present my work. Today I'm going to talk to you about the research management in an academic setting and how to set up a research center and manage it and what it takes to do that. Okay. I'll try to go through step by step, and then we'll see how it goes. When we talk about the research centers, the first center that we can think of is the CCMB in Hyderabad, which is a center for cellular molecular biology. But if you look at it, it has so many departments within the center. And this is a center that has been established by the CSIR, so I'm not talking about this kind of a center in a university setting. The second center that you can think of is the National Center for Biological Sciences, which is established under Tata Institute of Fundamental Research. Also has so many departments. I'm not talking about this. So we are talking about a research center which is an academic setting that we can do. Something that Indian Institute of Science has done, and which has done thematic research clusters, what they call it. And most of these things that you talk about, you see, they're all from artificial intelligence all the way to the project, 5G project. They are all in the technology sector. Nothing in the biology sector, but this is the technology center, and it is the Institute of Science Management that has decided that we need to have these research clusters in order to survive and succeed, and then in order to increase their research funding, and that is the way to do it, and they have established this kind of a thing. That came from the top down from the director of Indian Institute of Science. So, but if you look at the academic departments like any of the universities that we see, you see typical basic science departments ranging from chemistry to meteorology and oceanography. Okay. But at least in medical schools, it is different. In medical schools, most of the departments range from anatomy, biochemistry, cell biology, immunology, molecular biology to physiology. Okay. These are all the traditional departments that they have, and they have research within that cluster and whatever research that is done within that. But over the years, it has progressed that there is interdepartmental research, interdisciplinary research. Everything has become molecular genetic approach for everything. So how do we start a research center? In order to start a research center within an academic setting, one has to have an initiator who is willing to do this. Okay? So he must be a senior investigator, leader in his or her field, have a broad understanding of the overlapping fields, because you have several departments in a medical school or in academic in a typical university, and you should have an understanding of what is going on and what these implications are and what the long-term implications of this research would be. And they must have the time to spare. And finally, they need to have the skills to deal with the senior administration. If they're a good researcher and a good scientist, that's not enough. You need to have some sort of a business acumen to deal with the dean, to deal with the registrar, to deal with the vice chancellor, all that stuff. Okay. How do you establish a research center? Why do you establish a research center? Okay. when you establish a research center, I'm going to switch mostly towards the medical side and the biology side, because the engineering side we already talked about how Indian Institute of Science has done, but these principles can be applied to many of the other areas also. So in a theme-based research, so multifaceted research endeavors, they can be on one subject, and typically in a medical school it is a disease-oriented. This is the reason the United States medical schools have the maximum funding, because they're all focused on a disease-related research. So it can be in autoimmune disorders to substance abuse or thrombosis. Okay? It brings a lot of investigators with the common goals together. They all want to work towards one particular goal, say substance abuse. And you bring all the substance abuse people together and say that, okay, let's have a common research goal here. And when the investigators have different approaches to the same problem, some have pharmacological approach, some have biochemical approach, and some may have a zebrafish genetic approach, and they can do whatever they want to do, but they all have the same common goal. And it assembles all the resources in one place, because they all use similar kind of equipment, similar kind of resources, and you bring them together so they can flourish together in that setting. Okay. And the resources used by the investigators can be shared. Let us say somebody generates a knockout mouse, and that mouse can be shared by a different investigator because it is readily available within the center. Okay. So, given that, and now how do you set up a research center? For a senior investigator who knows what research is going on in the university, and they need to know, okay, in this department, this is the research that is going on, these are the researchers, these are their interests. Is there anything that is common from this department to that department, and can we bring them together? And that is the goal that a senior investigator has to have. And this person really should be motivated. Okay? Now you invite all these faculty that you think that they have a common interest, and then ask them to give their presentation and say, okay, talk about your research and let us see how we can bring together our ideas, and then is there a common goal that we can come up with. Now organize the seminars with a different possible themes. So you may bring in people from some departments on autoimmune disorders, or from some other department on thrombosis. And you bring them together and have the cluster of the seminars, and then say, okay, let's talk about this and then how we can set up this center. Okay. And you discuss the science as well as the possible thematic grouping. So you need to have a common understanding of that one. And approach the senior administrator with a solid plan. Now, after you know these are the investigators that are going to do this work together, and then now you go to the senior administration like the dean, registrar, vice chancellor, and say, hey, look, this is what we want to do. We want to set up a research center here, and this is going to improve the academic setting, so let's do the research here. Okay? And then the administration has to support you. And why the administration has to support you? Because everything has a financial implication. Let's say you don't have all these investigators. Can you build a center from scratch? You can build the center from the scratch, which is more difficult because then you need to recruit people on a common theme. That means you need to have the understanding and say, hey, I want to build a center on, say, inflammation. So inflammation research center I want to build, and that means you need to say, go to the senior administration, get the approval, and say, okay, let me bring in the people. Now you look into various people, various universities, and say, okay, we are going to recruit people every year, 2 or 3 people in the inflammation research, various aspects of inflammation research, and recruit them and then build the center. That's another way of doing it, but this is more difficult to do it when you do it from the scratch. Okay. The finances that are required for establishing a center are the funds that are required for the common equipment, such as centrifuges, micropretrators, to thermocyclers, to flow cytometer, anything that you want to buy for commonly, you need the money for that. Okay. And then you also need the money to acquire specialized equipment for various research things. Okay. And you have the administrative cost for the administrators. You need to have an administrator who is going to help you with the pre-award and post-award funding. You need to have the administrative secretaries who are going to do some of the work for you. You also need the infrastructure cost for modification of space, computers, internet, telephone, everything. You need to have the space that where you can conduct the research, right? And the cost for recruitment for faculty to the center, and interview visits to set up startup packages. So when you recruit people, and you need to pay for their interview, they are going to come from somewhere, and you have to pay for their interview, and the travel costs and all that stuff, and the money has to come from somewhere. And many of the places They give a startup package which is to start the lab, and that money has to be there somewhere, and that money has to come from the finances. And once you establish a center, and what kind of financial resources that you need the money from the institution which has to support you. And then you need to have overhead or indirect cost returns that will support your research center. And you have salary recovery, and then you have fund raise. Direct support from the institution is the salary support for the faculty. So in many of the universities, faculty salaries are paid by the university. So it is taken care of, and it doesn't have to be worried about, right? But the startup packages for the newly recruited faculty has to be obtained from somewhere, and that is the financial amount that you need from the institution. And the academic support that we talked about, the pre-award and post-award administrative support, that has to come from the institution. Now infrastructure, space, cold rooms, warm rooms, cell culture facilities, they all have to come from the institution, and the institution has to set up. They have to have the money to pay for that one. And then you also need the support structure, which probably exists in many of the places, like the Bay Telecoms, internet, and all that stuff. So that is already there. But it costs money to run the labs. The other way to get money for running the center is the overhead or indirect cost returns. So every research grant comes with an overhead cost or indirect cost towards the administration of the research grant. This money is not budgeted for the direct expenditures. Let us say you want to do research, and then you say this is the amount that costs me to do the research for the reagents, for technicians and the support staff, and then you need more money. So the more money that is coming is called the overhead, or it is called the indirect cost, which is given on top of the money. Okay? So the indirect costs in the US are based on the research infrastructure and facilities provided by the institution, and it varies based on the institution. For example, highly reputed universities like Harvard and all that, they demand a very high indirect cost rate, whereas smaller universities, they demand smaller indirect cost rate. So it varies. It can range from 40% of the direct cost to 150% of the direct cost. Okay. So in USA, the $1 million grant comes with a typically $500,000 for administrative costs, based on the assumption that you are getting 50% as the indirect cost rate. Whereas overhead costs in India are much smaller. So, towards meeting the cost of the overhead expenses, including the infrastructure facilities, this is the amount that is given. Typically, for a project costing up to 1 crore, 10% of the total cost of is given to the educational institutions. That is small, okay? So there is a deficiency in overhead in India compared to the USA. So a typical institution gets about 50% of the indirect cost from NIH in USA. And this translates to $500,000 in average research grant of $1 million for 4 years. This typical overhead is about 20% on a 10 million rupees or 1 crore grant in India. Okay. This translates to 20 lakhs rupees on an average research grant. The typical deficiency is about 30% in India on overhead. Okay. Why do you need this overhead? Because you are running a project and something happens suddenly. Your ultracentrifuge fails. Who is going to pay for the ultracentrifuge? Where is the money coming from? You cannot wait for the ultracentrifuge to be funded by next grant cycle or apply for a new grant. You have to have the money in hand. You go to the administration, and administration says, no, I don't have the money for new equipment. Okay? So for that, you need to have a buffer system that gives you the money. And skimping on the overhead. That is what is typically happening in many countries, including India, where funders typically support programs as pay for what it takes. Pay for what it takes means you propose, I want to do A, B, C, D. These are my specific aims in the grant, and this is what I want to do in the next 4 years. And you say, okay, what it takes? Oh, I need a technician, I need supplies, I need mouse costs, and this is what it takes. They give you that money. And they give a little bit more, which is the overhead that they're giving. Okay? You need to give more money. This practice gives rise to starvation cycle. The starvation cycle is because a lot of things happen when you're doing a research project, because something can go wrong, and you may have a new idea. Either way, whether an ultracentrifuge fails or you want to generate a knockout mouse in a hurry, because if you want to compete in the world of research, you need to have things done today. ideally yesterday, but today. Okay? So you need to get that done. How do you get it done? And you don't have any money for that. For that, you need the money. Okay? And the overhead given in India by the funding agencies thus limits the ability of the institution to grow their infrastructure and venture into the new research endeavors. That means when the institution gets the money, what do they use the money for? They use the money for electricity and all the infrastructure maintenance part of it, lawn cutting, whitewashing. Those are the kind of things that they're using the money for. Okay? But what they need to do is they need to invest money into the research. How do they invest money into the research, and where is the money coming from? That is the money that is coming from the overhead. So that means the funding agency has to recognize that if somebody is putting in a research grant that is worth funding, that means this is a place where we need to invest money. That is where the overhead or the indirect cost goes in. They say, okay, you are putting in a good grant, I'm going to give you extra money. This money is for the future, the way you can grow. And that same thing happens to the institution. Let us say institution comes with 20 grants. 20 grants it is supporting, that means, okay, your institution is doing well, so I'm going to give money in these 20 grants, extra money, that you can put into research and then you can grow. And that is where institutions can grow and make big impact. The indirect cost return in the USA is, the advantage of that is that some of this indirect cost that comes to the institution is actually shared with the investigators, with the unit. Okay? So the typical 50% of the indirect cost that we are talking about, 10 to 20% is returned to the unit, 10% to the investigator, and 5 to 10% to the school. That means if you are getting the money and 10% is given to the department or the research center that is doing it, and 10% is going to the investigator directly. And it says that, okay, 10% of this money you can keep it in your bank, in your account, and you can use it when you really need it. So if you have accumulated that money and then you want to say, hey, I want to generate this knockout mouse, you have the money ready to go. Okay? Or your grant cycle, you are running out of money and then you need to continue to maintain your technician because this technician is important. You have the money sitting in your indirect cost money, return money. That's the money that you can use. Okay? From that point of view, 10% goes to investigator, but if there are 10 grants that are in the unit gets a lot of money, and that unit can invest in research part. Okay? And the school gets 5 to 10% of the money, so school also can invest. Because there are 50 grants that are coming in the school or college, they can invest into the program. So in a typical research grant of about 1 million, there is about $500,000, but the unit gets about 50 to 100K, investigator gets about $50K, and school gets about 25 to 50K. And you can see that how much money is now accumulated by the number of successful people who are bringing in the research grants, and then the institution now is growing, and it is accumulating wealth. And this wealth can be invested into the programs. And when you go and ask for the senior administration, say that I want to set up this new research center, they have the money to give you. Otherwise, there's no money. So the universities will suffer. So cumulatively, the number of grants in the unit, in the school and all will have good amount of money, okay, for research endeavors. A research center that is established will be able to grow this by having new faculty and investing into the new core equipment and facilities, and also we'll be able to fund some of the investigators who have bridge gap, like they have a grant that is ending but the next grant hasn't come in yet, so to bridge them they'll be able to give the money and then support their staff during that. Okay. Lack of such indirect cost return in India is due to skimpy overhead. It hurts the institutions, the units as well as the investigators. Okay, the Indian government and the funding agencies have to put in more money into the institutions through a successful mechanism of the granting. The second part of that is the salary of the faculty. When salary of the faculty, when faculty are typically recruited, the entire salary is supported by the institution because the university salaries for the faculty are paid by the institution. The appointments are for 12 months. Then the teaching does not exist for the summer in most of the places. Okay? So the summertime there is no teaching, but they are just relaxed. Summertime is then the full-time faculty cannot carry the research but they cannot carry out intense research during this time. So if somebody can say that, okay, during your summer months you can do some research, but you cannot do research just by part-time summertime. You need to have a full-time investment into research. When the faculty obtain grants, then they can be relied on some of the teaching faculty to focus on the funded research. So they can actually say, okay, I don't want to do teaching because I have more research money coming, and now I want to buy out my time. They say, okay, I don't want to do that research part. But there is no additional incentive to carry out the research from this perspective. And the promotions are, of course, based on how many grants you got it and how many publications you have, but nothing more than that. But the salary incentives evolved in the US in a different way. So typically university appointments are for 9 months, okay, leaving the summertime for research. And some faculty began utilizing the summertime to carry out some research and then bringing in the money by applying for grants and supporting their salaries. Now faculty and departments resisted this idea of putting the salary support on the grants. Only for the summertime they were getting it, but remaining time they didn't want to put it. But slowly, buyout concept emerged where the universities and departments said that, okay, if you bring in the grants, you support your salary, we are not going to ask you to teach. You can be relieved from teaching. So this is the buyout. And this is significant, as the full-time teaching load is about 12 credit hours per semester in many universities. That's a lot of teaching load. So 12 credit or contact hours means it's a lot of teaching load and you don't have any time for it. Seeing this new opportunity, schools started pushing faculty to put grant money and support their salaries. For that, for putting the budget in their salary and grants, now they started giving them incentives. How do they incentivize them? So one of the ways is that as the salary of the faculty is already accounted for the university budget, so they want to give incentives. So in order to encourage them, so they give more money in the salary for that, so they started salary recovery incentive. Salary recovery is that as a percentage of the salary that comes back to you, now you say, okay, I'm going to give part of the salary that you brought in to the unit and also part of the salary to you. And the salary that is given to you is more like the indirect cost return where you can use it for research purposes. So this incentive will encourage faculty to put more effort on the grants and bring more money towards the salary. So there is an incentive that I want to put more money, I want to get more grants, I want to grow is the drive that comes from our faculty. Instead of saying that, okay, university is paying my salary, I don't have to worry about it. Typical incentives for the 20% of the money is within 20% of the money will go to the investigator. That's the money that counts. And they also have the salary increase. So the salary increase typically in this case is about 35% of your salary is taken care of, but anything that you bring more than 35%, you actually get an increment in your salary. Okay? That is the salary incentive. So you get that money into your bank account basically as a salary increase. So it can get up to 20% if you are a really successful investigator. You will get 20% of your salary into your account. So that is a big increase. And the success of the research centers, okay. Their successful faculty will bring additional money in the form of indirect costs, salary recovery, and they can increase the research funding by multi-investigator grants like the program projects, and they can write large grants, bring more money to the institute of the center, and that leads to a research center to grow because they are bringing in more money, they are banking more money in their costs. And you can also do the fundraising of the center. So you can go to the wealthy philanthropists and say that, okay, I want to name my center in your name and give you money. Okay. Some of them, they have some sort of an association with the disease and they may say, okay, somebody in my family died with this disease, so I'm going to give, this is the research center, I'm going to give you money for that one. You can also ask people to contribute to this, like diabetes research, and people may contribute small amounts, 100 rupees, 1,000 rupees, they may contribute at the checkout, and they say, okay, I'm going to give it for the diabetes research. You may grow money that way. And you can also go into your alumni and say, who are successful, and say that, okay, can you contribute to the growth of the research center? And they may give money to that. University faculty appointments can be reduced to 9 months, saving 2 months of the salary. This is the translation of how we can apply to the Indian context. Granting agencies can allow for salary support on grants, so the institution can divert the salary money saved towards research. This is the recognition of a successful research from an institution and reward to the institution in the form of the overhead. Granting agencies can increase the overhead by provide additional developmental support to the institution, a reward for being successful in research funding. Such bonus will help research infrastructure and institutions and makes them better equipped in the future for the successful research. Granting agencies can award large grants such as multi-investigator grants, multidisciplinary approach towards a common theme, and such research collaborations and multidisciplinary approach reduce internal rivalry and enhance common good. Okay. These are several research centers that are established within Temple University, and you can see some of them are named as Blue Cross Blue Shield because they gave the money, or the Lemol Center. The Lemol gave money for the research center, and they can establish, and there is an endowed chair that they can establish. They did that. Okay. In conclusion, enhancing research capabilities and making significant contributions to requires large-scale investments into research and development. In academic settings, granting agencies need to provide more funding towards overhead instead of limiting the funding to pay to work done. Academic institutions have to find ways to incentivize the faculty to apply for extramural funding with significant salary support. And research centers must be formed with a thematic purpose to enhance collaborations among investigators and submit large-scale research grants. Okay. Thank you.
Thank you, Dr. Kunapalli, providing us how to initiate the centers and the various costs that needs to go and consider while establishing the centers and comparing from what is done in India as well as in US. You can have a seat here, so we can take the questions later. Now I invite the next speaker, Kumar Ralapalli. He is the global expert in vaccine and public health. He has already spoken, so I'm not going to give the introduction again. So he can start his presentation on key drivers for the successful commercialization of biotechnology and pharma industries.
Okay, thank you very much. I promise I will be brief and fast. Okay, all right, thank you. Hi everyone, hello again. So this part of the presentation is closely related to what Rohini presented earlier. So I'll talk about how companies make decisions on which products they want to invest in and how they bring them to the market. Okay, let's do it. Pharma industry, it's typical, like any other industry, is going through a major transformation. There are quite a few forces that are operating right now. Everyone's trying to figure out what to do post-COVID. Pharma actually has been going through quite a bit of change in the last 10 years, shifting tremendously from primary care medications to specialty medicines to rare disease to vaccines, and so that'll continue. There are new modalities, and there's pressure to innovate. So these are all the factors that's going to affect pharma. So how do they make their decisions? So it's fairly well accepted framework, if you will, that companies go through in terms of identifying where they want to invest for their next asset to bring to the market. I'll go through those 4 big steps. Those are displayed on the screen: unmet medical need, technical feasibility, regulatory pathway, and commercial opportunity. If I just double click on the unmet medical need, it's how do you know there's an unmet medical need? Well, obviously, if you're looking at the literature, if you're speaking to the stakeholders, if you're speaking to healthcare practitioners, typically you would know. So there's usually quite a bit of evidence on potential disease areas that companies should invest in. The other key term that companies use for unmet medical need is what you call disease burden. Typically, you look for the metrics that suggest what people die from. Cardiovascular disease is the #1 killer worldwide, #1 killer in India. If you look at top 10, then you'll have cancer, infectious disease, others. So these are things that you look for. You look for morbidity, mortality, dailies, disability adjusted life years, incidents and prevalence. Those are the typical metrics that you look for, and then you try to address based on what your capabilities are, depending on if you want to pursue a small molecule or a vaccine or a monoclonal antibody, things like that. Once you go past, okay, we're going to go into an infectious disease and we're going to develop a vaccine because there are a lot of people that suffer from dengue. Okay, so dengue is a global, there is a global disease burden for dengue, and so you could develop a vaccine. Then you need to figure out how, if it's even possible. So if it's possible, we would have a vaccine for everything. Then we wouldn't have any disease at all, right? But it's not possible. So that's the technical feasibility part. And so you look for ways to develop an antigen that can be safely administered, and then of course has the efficacy necessary to protect you from the disease. And so there's quite a few vaccine technologies that are available now, more so than ever. And so that's what you will go through in terms of the technical feasibility. So the mRNA is the new kid on the block. The proof of concept is the COVID vaccine. Now all potential disease targets and infectious diseases, even in cancer, they're looking at mRNA as a potential platform. So that's a classic example of a new technology. You're trying to figure out the technical feasibility to get to the end zone. Once you have that sorted out, obviously you have to figure out the regulatory pathway. This is usually through consultation. Obviously every country has a well developed regulatory framework. You speak to the National Regulatory Authority. In India, of course, that would be DCGI. In the US, it would be FDA, so on and so forth. And so typically they would work with you in providing the consultation on how to get it through. And last but not least, if you're a private pharmaceutical company, then you are interested in the commercial opportunity, simply because that's the way you sustain your business. It's not charity. If you're an NGO, you don't need to worry about commercial opportunity. You, of course, still need to worry about fundraising, but for a pharmaceutical company, you need to worry about commercial opportunity. And so typically companies would then look at the disease targets and then say, maybe we're not going to focus on TB. We're not going to be able to generate a lot of revenue, but maybe dengue, because we can market it in many countries. There may be greater revenue potential, so let's go after that. So that's sort of the thinking. So you look at the substantial size of the population based on number of people that will take this medication, and then you also look at the competition. If there are 10 other people developing similar medications, then you're not going to pursue that. Okay. And then the key term that companies look for before they develop anything is called target product profile. So you map out the product and the way you want it to look like. You want a once a day medication. You want it to have 50% efficacy. You want minimal side effects and things like that. This is your ideal profile. And then you do the tests, and you will probably never achieve the ideal profile. And so you'll look at how far you are from the ideal profile and see if it's still acceptable in the marketplace, and that's what you market. So just to quickly summarize the way companies invest for new therapies, vaccines, pharmaceuticals, they'll look at the unmet medical need, see what the burden is, and see if they can target some of the diseases. Do they have a pathway? Do they have a technology? Do they have expertise? Do they have a pathway for regulatory? And does it pay back? And so if you have all those checks, then the companies are going forward. There's two other steps, of course, to get the product to the market. One is you develop the product to bring it to the market, and that's what we call prepare the product for the market. And so these are all the steps that you do: clinical development, regulatory approval, manufacturing. And then and then the second part is you prepare the market for the product. For example, if you develop a product for dengue vaccine and then you show up, it'll take a long time for you to sell that product, because then you need to tell everybody, here's a new dengue vaccine, go to your doctor, get a dengue vaccine. So what companies do is about a year before they actually bring the dengue vaccine, they will start communicating to the stakeholders that there may be a dengue vaccine coming soon. Company X is working on dengue vaccine. You're going to see all this news everywhere. So people are already mentally getting ready. And so that's what you call preparing the market for your product. First you prepare the product for the market, but before your product comes to the market, you prepare the market for the product. And so the way you do that a lot of times is by speaking to the stakeholders, talking to the patient advocacy groups, but most of that has to be scientific. So you cannot promote a product before it is approved for licensure. So you don't see a company go out and tell, we're going to come out with this new dengue vaccine, it's going to be great. Nobody will say that. So you will see companies being very responsible in saying, we are working on a dengue vaccine, we anticipate the study to be completed by XYZ date, and we are hopeful that the vaccine will be safe and efficacious. That's the language. But then the news is out, right? Okay. And then that's it. I'm just going to wrap it up. I'm sure all of you are ready for lunch. So yeah, that's it. That's the two key areas. One is the asset identification process. You go through and select the product, and then you make it ready for the market by developing the product first, preparing the market next. And with that, I conclude.
Thank you, sir. Thank you so very much for your brief but informative talk. Yes, ma'am. Now, I request, okay. Now, if you are having any queries or any questions, please you can ask these speakers. Participants? Yes.
Pharmaceutical companies, because it's tied to the project management topic that she's talking about. So typically, pharma it's funded on their own. So it's coming from their own investments, and that's the reason they expect a return on it. In case of COVID vaccines, they didn't fund it on their own. They actually got quite a bit of support from governments all around the world. And therefore, one of the things that they had to do was to manage the pricing. So they couldn't price it as they wanted. The governments had a very close agreements with them to make sure that the pricing was well controlled. So depending on the disease target, if you found a pharmaceutical company, I am looking to fund it on my own, depending on what stage the development of the disease target is. But early stages, most of the pharma innovation is not in-house. So I think there are others that talk about academic research centers. Typically is where the early research starts. Pharma usually takes it from there through either a licensing or a royalty or something like that, and then that's where the funding comes from. Yeah, sure.
Any other participant? Yes. Speak loud.
That's a good question. Yes. So what's the difference? I'm trying to think. Obviously, IP is critical too. So I'm going to keep it as generic as possible. just, IP is critical. In terms of difference between India and US, I think obviously you're worried about having a robust legal framework to protect your innovation, and so I think that would be the overarching concern if I'm a global pharmaceutical company to go anywhere, not just India. You don't have to worry about India. I think India is certainly it's part of the TRIPS agreement, and it's playing by the rules, so to speak. So that's that. I don't think there's any potential concerns, but that would be the one that weighs on any company, whether it's American or from any company. If they're planning on entering India, IP would be something that they would take a look at, and I know that that's potential cause for concern. But I think we are in 2022. I think we're probably looking at pretty much every company looking at India as a viable market that they can bring their new products to. Perhaps maybe a while ago it would have been a bigger concern. I mean, you're closer to this than I am. So.
I can chime in from the project management side, because I also work with alliance teams where they begin the contract, because my company have a collaboration with Serum Institute here, and we screen legally all the documents that comes in as a contract form to make sure that there is no intellectual property. All the claims are all like very clear. Those contracts will take like months to get aligned between the companies, and it's a long process, and very diligently reviewed by both parties. Still, there will be some conflicts that will come in, but again, there will be like a legal team fully engaged in those things to cover both parties.
Isomer from the under consideration, and the same route was done with 6-membered cyclic thiourea. So our aim was to study their DNA interactions, whether they are minor groove binders or they are intercalators or they cleave the DNA. So we carried out molecular docking study with a dodecamer for both the series of compounds. And it was interesting to note that these molecules bind with guanine, cysteine, ring. Also interesting to note that Lipinski rule of 5 is well satisfied by all the compounds. And from 10 compounds from each series, we selected one single compound which shows the best activity, that is having 2,4-dichloro substituents. So everything in the molecule is same. The only difference is a 5-membered imidazole ring or a 6-membered pyrimidine ring. So we wanted to study the effect of ring size with the with the DNA binding. So both were binding well in the same minor groove region, and compounds have high drug likeliness, promising drug score, and low toxicity, which was calculated using Osiris program. So these are some of the measurements which we have done, which confirmed that the molecule is binding. Absorption, UV absorption spectra and fluorescence spectra clearly indicate that when we add our ligands into the DNA in UV, there is increase in absorption, and in case of fluorescence there is decrease. So for these we were confident that these compounds were binding with the DNA. Then the question was whether it is intercalating binding or it was minor groove binding. So we did the competitive studies. So when the study was done with the Horchest 335258, which is a well-known minor groove binder, there was replacement. So that shows that our compound is binding in the minor groove. And when we tried to compete with ethidium dibromide, which is a good intercalator, established intercalator, there was no exchange. So from there we got the idea that the compound is minor groove binder. And CD spectra also confirmed that, because CD spectra did not any change. So that confirmed that the compound is binding in the minor groove. Similarly, viscosity measurements supported our observation that the minor groove binding is taking place. So the same compounds were also explored for BSA binding, BSA, bovine serum albumin and human serum albumin, they both are having the same analogy, and they are, the mobile protons in the circulatory system. So it's very important molecule BSA to study on. So we've taken up binding studies, and it shows that the docking score for imidazole series was minus 8.3 kilocal, while for pyrimidine series it was minus 8.0 kilocal, and they are binding in the pocket where arginine and threonine, alanine, etc. are there. Again, absorption studies, fluorescent and UV absorption showed us that they are interacting, and job plot confirmed that they are binding into 1:1 ratio, and unbinding of alpha helix takes place. These were established by UV absorption studies. So by comparing, we can easily say from the docking results that both the ligands alter the absorption and emission spectra of DNA and BSA. With DNA and BSA both, magnitude of quantum and Stern Volmer constant suggested that 5-membered fused ring system binds DNA and BSA effectively. But the overall observation, if you look at the binding constant for DNA for imidazole series is 1.4 into 10 raised to power 4, and for pyrimidine series this is 3.9 into 10 raised to power 3. So that clearly indicates that by increasing the number of carbon chains in the fuse system decreases the binding ability of the molecule with double helical DNA and BSA. As I said earlier, that CF3 trifluoromethyl group is a very important group because it is almost similar in size with CH3 group, so it is safer to add it. But CF3 group, fluorine being the most electronegative element, when it is introduced in the molecule, it changes lots of properties, and the biological profile is quite different. So our aim is to synthesize corresponding trifluoromethylated analog, but to our surprise, we got the difunctionalization of nitrogen and sulfur, N-acylation and S-alkylation in one step. Again, the characterization was done by 2D nuclear spectroscopy, and I must add here that there are not many methods available to synthesize such kind of compound where nitrogen and sulfur both are functionalized. And another interesting part here, the carbonyl group exists in the dihydroxy form. So that means there are more opportunities for hydrogen bonding with the biological molecules, and it's acyclic structure, therefore more flexibility, to fit in the binding pockets. Another interesting fact from the docking score, I told you in the last slide, the cyclic molecule was showing the docking score of minus 7.1, or in BSA minus 8.0. But in this particular series, by replacing CF3, our binding score has gone to minus 7.9 and minus 8.7. So that means these are the compounds which has much better ability to bind with DNA and BSA. The mechanism proposed is a free radical mechanism, where we are suggesting that S-alkylation takes place first and then the intramolecular cyclization. But in the case of trifluoromethyl substituent, instead of intramolecular cyclization, CC bond cleavage takes place regioselectively, giving only one regioisomer. The free radical mechanism was supported by free radical squenger and free radical initiators, which clearly indicated that the reaction is progressing through free radical mechanism. It also encourages us to explore acyclic thioureas. Acyclic thioureas, because these are monosubstituted, so both the nitrogens have role to play. At this time, we know that sulfur, being the more nucleophilic, attacks first. Therefore, we eliminated the possibility of two regioisomer. But here we have to consider the second nitrogen, endonitrogen, which can also participate. So again, 4 possibilities. Out of 8, I am writing only 4. So one is amino substituted thiazoles, the other two are 2-amino N-substituted thiazolene derivatives. So initially the reaction was carried out in sodium carbonate and solvent-free reactions. Interestingly, we got, this reaction was done on more than 27, 27 reactions were performed having different thioureas and different diketones. So in some cases we got the desired thiazoles, amino thiazoles, but in some cases we got the exclusive formation of not the expected aminothiazolene but a rearranged product where acyl group has migrated from carbon to nitrogen. A very interesting pattern of allylic coupling, that CH3 appeared as a doublet because of the thiazole present at position 5 and a doublet for hydrogen at position 5. So this is only possible if the acyl group migrates from carbon at position 5 to nitrogen present at position 2. Both the molecules, because the new molecule you are doing the 2D NMR spectroscopy, but just to confirm it, we went for the crystallography. We grew single crystal of both the isomers, which clearly indicates that the assignment is correct. And again I would like to mention that aminothiazoles of this kind, only a couple of methods are reported which are multi-step. Here we are getting in one step CNL acyl migration, and this is an intramolecular mechanism through a 6-member transition state that the group migrates, because once the thiazole ring is formed, it's almost impossible for nitrogen to attack the acetyl group and migration takes place. We have done the crossover experiments where only 2 compounds are formed, not one. So that indicates the reaction is intramolecular. Interestingly, as I said, visible light may give us different results. When we carried out the reaction in visible light, only exclusive product 5-amino thiazole derivative was formed. So that means visible light is changing the route of reaction. Then we went to thiamides because there is sulfur and nitrogen. The other nitrogen is missing. When the reaction was done in solvent-free condition, we got the expected product, because now we are much, quite confident that the acetyl group is at position 5 and methyl group is at position 4, proved by X-ray crystallography. Interestingly, when the reaction was carried out under visible light, oxidative dimerization of thiamide occurred. We couldn't find any trace of diketone, and this is a method, very green synthetic approach towards synthesis of 1,2,4 thiazole. The reaction yields are 80 to 90%, and the mechanism shows that how the oxidative dimerization takes place, because there is formation of S-bromo compound which makes sulfur highly electrophilic, and the second mole of thiamide reacts with it and followed by cyclization gives us the thiazole. 2-amino pyrimidine, 1 nitrogen internal, 1 external. Again, the similar kind of product was obtained under visible. However, under visible light, we could not achieve any product. This was done by DCM route, and the X-ray crystallography confirmed the structure. 2-mercapto 1,2,3 triazoles. Again, only one isomer was formed. The structure was established by 2D and crystallography. We extended the reaction to trifluoromethyl beta diketones, and here you will notice that again an acyclic product has been formed rather than the cyclic product, and this time the different regioisomer has been formed. Earlier we have COR group at nitrogen and COCF3 group at sulfur. So the regioselectivity is changing in this particular case, and the compound was confirmed by mass spectroscopy and NMR. And another interesting thing is that in this case, dehydration has occurred, and instead of dihydroxy, it exists in the carbonyl form. We have amino mercaptotriazoles, thiodiazines were formed. Structure was characterized, and when the reaction was carried out with trifluoromethyl beta diketone, interestingly, COCF3 group gets cleaved. So, while at one hand we are confident that when the reactions are being carried out with trifluoromethyl beta diketones, we get most of the time one product with some rearrangement, some with some oxidative dimerization. With trifluoromethyl beta diketones need further investigations, because every time we are getting a different product, and which is quite interesting to study. The second part is the multi-component synthesis. You all know the uses of this, so therefore I will not emphasize. So I will take couple of reactions in each class. So here the reaction of heterocyclic hydrazines with diverse 1,3 dielectrophiles. So synthesis of pyrazolo 3,4-B pyridines. So two isomers are possible, 5-amino and 3-amino. Initially we carried out the reaction in stepwise mode. Hydrazine reacts with beta ketonitriles to give us 5-amino pyrazole, which reacts with trifluoromethyl diketones, giving us a single regioisomer 4CF3, which was characterized along with the presence of a side product where an acetylation takes place. The reaction takes much longer time and irritating solvents are used. Therefore, we thought to go for multi-component synthesis. Interestingly, the isomer obtained in this case was a totally opposite. Here we got the 6-trifluoromethyl compound, and it was easy workup, milder reactions, enhanced reaction rates, high level of regioselectivity, and we have both the isomers to study their biological properties. Going further, we have observed that when there is possibility of two regioisomers, and if we change the conditions, many times the second isomer is formed. So we wanted to carry out the reaction of hydrazine with beta ketonitrile in presence of acid. It was, an AMPhil student who was working on this problem, and generally for AMPhil students we look for a very, simple problem so that they can finish it in time. Accidentally, the girl used ethanol, which was contaminated with nitric acid. So she used HCl a drop, but the ethanol had the nitric acid. And I must share with you, there are long stretches when we do not have NMR facility, in our university. So what we do, we keep on carrying out the reaction, we synthesize the products, and then, when a batch is ready, then we send it for the NMR. It was very surprising that every compound was showing a doublet and a quartet. Had it been in one compound, we would have missed it, like koi impurity aa gaya hoga. But every compound, a very beautiful crystalline compound, showing a doublet and a quartet. And looking at the composition, we found that there are two molecules of beta ketonitrile which are getting integrated in the molecule with hydrazine. But interestingly, there are two additional carbons. These two additional carbon cannot be from hydrazine, cannot be from beta ketonitrile. The only source which can be, that is ethanol. And then we proposed that ethanol got oxidized in situ in presence of nitric acid, and then the integration has taken place. So we wanted to go for the X-ray structure. We grew single X-ray crystal, and when we sent it for the X-ray crystallography, we got the structure which is a totally aromatic system. NMR of this compound does not show any doublet quartet, and it shows mass spectroscopy shows 2 units less. So that means while crystallization, aerial oxidation has taken place, and dehydrogenation of the dihydro product has happened to give us the aromatic product. And this compound, though does not have any chiral center, but it showed 2 enantiomers because the 3 phenyl groups attached on the pyrazolopyrimidine are propeller shaped, which give the dissymmetry to the molecule. Utilizing this, we have synthesized a large library of these compounds which are biologically very active, and we have used even different beta-ketonitrile, how we can bring the diversity in the molecule, and instead of only ethanol giving us acetaldehyde, we have explored a large number of aldehydes into this. And looking at the mechanism, we are proposing that acetaldehyde is produced in situ, in domino reaction occurs, and then it combines with beta-ketonitriles novenigel reaction, and further condensation gives us this product. Again, I would like to mention here that this method to synthesize dihydropyrazolopyrimidine is not a well established route in the literature. Coming to the green catalysis, while at Cambridge and at TSA University, I had the exposure for biocatalyst or thioesterase and antibody catalysis. But here, because we are talking about aza-heterocycles, there is one marine product, basilamide, which was synthesized earlier by six-step method, and the overall yield was only 14%. It's a very selective algacidal compound. And today we are all talking about, marine natural products. They are very unique. So we wanted to do the synthesis, very simplified synthesis, just in 2 steps. So first problem was to construct a thiazole ring with the right substituents, and the second thing was to synthesize, to develop a method for amide bond formation, because the earlier methods used the mixed anhydride method, giving you poor atom economy. So we started with cysteine ethyl ester hydrochloride and pyrovaldehyde, in situ oxidation with iodobenzene diacetate, that gives us the right substituent at position 2 and 4, and further condensation with tryptamine in presence of 2-hydroxy-4,6-dimethylpyrimidine gave us basilamide in excellent yield. So overall yield of the reaction is now 42%, and from 14 to 42%, and the beauty of this reaction is that we can include diversity in the basilamide molecule. So instead of using pyrovaldehyde, we have introduced many aldehydes, and at our position we have several groups. The mechanism what we propose, that this catalyst provides hydrogen bonding, bringing the ester and the amine closer by bonding simultaneously with both the molecules. So these compounds were evaluated for their cytotoxic study, and it was interesting to note that the analogues are better cytotoxic agents against colon cancer, breast cancer and leukemia cells as compared to basilamide. Now coming to another catalyst which we have used very nicely. For last 3 decades in our research group, we have been working with the reaction of hydrazine with trifluoromethyl beta diketones to incorporate trifluoromethyl group into the pyrazole ring, and salicylic oxide is the molecule which have 3 CF3 group in it. But every time when we do the reaction, 5-trifluoromethyl or 5-hydroxy 5-trifluoromethyl pyrazolene is the major product. Here also, it started, I have extended this work from, Dr. Singh's lab, because I have taken the legacy from him. So we have done more than 60 reactions to establish that as we increase the electron withdrawing group nature of the substituent, pyrazolene is formed. When we increase the substituents on the hydrazine, pyrazolene is formed. The only thing is, when we increase the nature from neutral to acidic, trifluoromethyl is there, but it is never an exclusive product. So looking at the salicylic oxide, which is having 3 CF3 group, we were in search of a method which gives us exclusively 3 CF3 derivative without a trace of CF3 or hydroxy CF3 pyrazolene. So our target was hydrazine pyrimidine because it has the same profile as the sulfonamide group. So we carried out the reaction. Again, all the 3 products were formed. We carried out the docking studies, which clearly indicated that 3-trifluoromethyl isomer is a better analog of salicoxib as compared to 5-hydroxy. So therefore, we take the lead from our earlier studies where we have synthesized carbon-nitrogen bond, amide bond, using aliquot 336 as phase transfer catalyst. So we have, developed an alternate route. We condense trifluoromethyl diketones with hydrazine, which gives us unsubstituted pyrazole, which has the ability again to give us 2, heteroaryl hydrazine through aromatic nucleophilic substitution. In this case, we use aliquot 336 as a catalyst in presence of an inorganic base sodium hydroxide, and heteroaryl chloride were added, which are suitable for nucleophilic substitution. And interestingly, only single isomer 3CF3 is formed. So now we have an alternative route to achieve a salicoxib analog having 3CF3 group. Solvent-free synthesis, again, we are not using any solvent, so it's eco-friendly route, where we have 2-amino 5-hydrazino pyrazoles. Reaction with beta-ketonitrols give us, the product where the hydrazine gets cleaved and we get pyrazolo 1,5-A pyrimidines. Very different reactions occur when we do solvent-free synthesis. This is another reaction where pyrazolo thiocarboxamide on reaction with alpha haloketones lead to the formation of alpha thiocyanate ketone because of the CN bond cleavage, and in situ formation alpha thiocyanation occurs. But when the reaction is carried out in sodium carbonate solvent-free grinding conditions, this CN bond cleavage can be avoided and the hybrid molecule of thiazole imidazole can be achieved. Coming to sustainable solvents, we have used water, which is known as the universal solvent and the best solvent. No toxicity. So we utilized on-water reaction, and very interesting products were formed, giving us high level of regioselectivity. And these compounds were evaluated for their antimicrobial, antifungal activities. The mechanism was studied in deep. I am skipping that. We have also synthesized some compounds having the 3CF3 group and thiophene ring, and these compounds showed very good cytotoxicity against cancer cell lines. Also, very interestingly, these compounds are very good photocleavage agents because they cleave the supercoiled plasmid into the smaller fragments of 20 micromolar. So both the activities, DNA photocleavage and cytotoxicity, makes them good candidates to develop anti-cancer agents. Lastly, green reagents, we have been using hypervalent iodine as green oxidizing reagent in our lab. The two main iodine 3 reagents which we are using is iodobenzene diacetate and hydroxytosiloxy iodobenzene. These are oxidizing agents which are eco-friendly, easy in handling and non-toxic. In the earlier slide I mentioned that we are having some murderous elements which we use quite frequently in our lab, which are thallium and lead and nickel, etc. So iodobenzene acetate, iodine 3 compounds basically have the similar mechanism and similar ability, so we can replace heavy metals with iodine, which is safer. So utilizing this, we have synthesized quinoxylene N-oxide from 1,2 diketones, making the oxime and then Schiff base, and it condenses to give us oximino Schiff base, and intramolecular cyclization with IBD, the mechanism written here, gives N oxide in excellent yields. The same approach of iodobenzene diacetate as an oxidizing agent has been utilized to synthesize a large number of fused triazole system. Here I am showing only the one here where we are having taken pyrazine. It reacts with hydrazine to give us corresponding hydrazine. With aldehyde we get hydrazones, and hydrazones intramolecularly oxidize to give us triazole. The reaction was repeated again and we got bis triazoles, which are excellent compounds in their biological activity. Photocleavage DNA activity of these compounds have been done and they have shown significant reactivity. My last slide of hydroxytosyloxyioidobenzene. This molecule is very useful to synthesize alpha-tosyloxyketones, which are eco-friendly synthones equivalent to alpha-haloketones, which are lacrimetry in nature, unstable and difficult to synthesize because they use organic bromine. So here we synthesize alpha-tosyloxyketones by reacting enolizable ketones with hydroxytosyloxyioidobenzene, and they can be generated in situ. You don't have to do the isolation step. And reaction with thioureas, we have synthesized under different reaction conditions amino derivatives and immunoderivatives. Similarly, these compounds have been utilized in one step to synthesize a diverse kind of pyrazoles, thiazoles, imidazoles and fused and heterocyclines. Some of them I have shown in the picture. So with these I would like to end my talk by acknowledgement. As I mentioned, whatever I am today, I think it is because of the guiding force, Professor S.P. Singh. I got the opportunity to work at different labs at Dr. James Staunton at Cambridge University, Fabio Benedetti at the University of Trieste, Professor Jonathan Spencer, Cambridge University when I was a Commonwealth Fellow, and Professor Isabel Rosas, who is at Trinity College Dublin. And my collaborators, I must thank them because many of the times we did not have the high-end facilities available at Kurukshetra University. So they very generously worked with us, collaborated with us, Professor Clarament, Professor Sanz, Professor Duro, who is known as the father of pyrazole chemistry, all from Department of Chimica Organica unit, and Dr. Rachna Sadana, who is very closely working with us on the cytotoxic studies, and I am very glad that today she is there with us and delivering plenary address tomorrow. PhD students, I think they are the backbones of any researcher. My past and present PhD scholars who have worked with me day and night. There were many times when we were not having the facilities required immediately, but they have done wonderful thing. Whatever publication score I show, it's only because of them. And I must say, for the last 3.5 years that I am away from Kurukshetra University, I am at Delhi. My research group is, I think, performing better than I was there. So they are so brilliant, I can say them. Particularly, I would like to mention Dr. Garima Sumran, Dr. Mona Huda, she has submitted her thesis, and Mr. Prince, who have helped me in preparing this presidential address, and Suraj. He is a project assistant at CSIR Whisper. The day Indian Science Congress was announced, he is working day and night with me. Many of the emails you have received, Suraj was the man behind it. And not forgetting the funding agencies, Association of Commonwealth Universities, University Grants Commission, DST, CSIR, Haryana State Council for Science and Technology, for generously funding us time to time, so that our research group is always having sufficient funds, not looking for. And lastly, but most importantly, I would like to thank my parent organization, my alma mater, Kurukshetra University. The land of Bhagavad Gita, I can say the first university of the universe, a beautiful campus sprawling over 500 acres. You must visit this place, it's a wonderful place. And CSIR, Council of Scientific and Industrial Research, particularly my institution, National Institute of Science, Communication and Policy Research. It's a new name, many of you may not be knowing, we are only 2 years old because last year it was created. But this institution has the legacy of more than 100 years. CSIR NISCARE, which was established in 1942 in different names, along with CSIR. So it is having 80 years of legacy in science communication and information resources. All the e-resources which you get just by click on your laptop, that is by National Knowledge Resource Consortium, and we are the one who are working on it. And CSIR NISTATS, which has the legacy of 40 years, which is working on the science policy, giving advice to the government and CSIR, and both the institutions basically work at the interface of science, technology and society. You are again welcome to CSIR NISPA 2 at New Delhi. So with these words, I would like to once again thank you all and my gratitude to Professor Tiwari, Professor S.P. Singh and other lovely members present. Thank you so much.
Friends, we have had a very interesting and illuminating talk from Professor Ranjana Agrawal. You must have realized how she has been playing with the molecules and creating different molecules. It's an art. I don't know how she has managed to do it. It's very difficult sometimes to link two things. She has linked so many things. These words, I think it's time we should appreciate her lecture. Please join me in appreciating her lecture. Thank you.
May I request Professor H.P. Tiwari sir to felicitate Professor Ranjana Agrawal madam with a memento as a token of love, respect and gratitude. Thank you sir. Thank you ma'am. I request Professor N.T. Khatti, Professor Department of Chemistry, Lakshminarayan Institute of Technology, to propose a formal vote of thanks.
Good afternoon, Honorable Tiwari sir, Honorable Ranjana Agrawal madam, all dignitaries sitting over here. I am formally vote of thanks. So I wholeheartedly give my vote, my thanks to Professor Tiwari for chairing this session. I am also very much thankful to Dr. Ranjana Agrawal, Director NISCARE, for presenting here a presidential address and giving very beautiful information about various steps of the organic reactions and the green synthesis of that organic reaction. I am also very much thankful, Madam, for supporting me in organizing this chemical science section along with the Chavan sir who are supporting me day and night and always giving me support for everything what is required for organizing this event. So really wonderful session by you, madam. Thank you very much. Thank you.
Today we have with us another distinguished and esteemed personality from the field of chemical sciences, Professor S.P. Singh sir. May I request you sir to please occupy a seat on the dais. I request Purnima Agrawal ma'am to please welcome Professor Singh sir with a floral bouquet. Thank you ma'am. Professor S.P. Singh sir, a brief introduction about him. After obtaining PhD from Lucknow University, Professor Singh sir has joined Kurukshetra University in 1965 and served the university as chairman in Department of Chemistry. On his superannuation, he was appointed as Professor Emeritus for the university for lifetime. Professor Singh has worked in various areas of organic chemistry and has published about 200 research papers and review articles, besides supervising a large number of students for PhD degree and also authored many books. He had spent about 5 years in the University of Illinois at Chicago as postdoctoral fellow, Fulbright scholar and visiting research professor. He was INSA Royal Society Fellow to University of Bath, UK twice and spent some time in CSIC Spain. He was elected President of the Chemical Sciences of Indian Science Congress and later as the General Secretary for a 3-year term. He is now permanent member of the Council of the Science Congress. For his services to science, Professor Singh was honored by the Prime Minister of India in 2014. He has also received the Lifetime Achievement Award of the Indian Chemical Society. May I request you, sir, to please chair the session.
Where is he hiding? Come on, I mean, we should put a name plate for him. I hope he's not occupying my chair. Well, I'm very happy to introduce to you Professor Rawat, who is my friend for the last 20 years. I think he's among one of the most bright young organic chemists with whom we pin the hope that he would contribute something great for Indian science, particularly chemistry and organic chemistry. Professor Rawat, many of you may be knowing about him, but just a brief introduction. He did his MSc from the University of Kumaon, and after that he joined CDRI Lucknow and worked with Professor D.S. Bhakuni, with whom I had also a chance to spend some time when I was doing my PhD. After that he moved here and there and then joined University of Delhi, where he is currently a senior professor of chemistry. Many of the areas in which he had worked, I know all the areas because I have been associated on academic ground with him. They concern about finding new molecules as anti-cancer or antimicrobial, and now more importantly anti-Parkinson. For Parkinson disease, you have got recognition, global recognition, to discover some molecules which may against Parkinson, which remains to be a very big challenge for the mankind. For his work, he has been recognized well. He is fellow of the Academy or National Academy of Sciences and have got many awards. I can say this Indian Science Congress, R.C. Shah Award, and last session, he was the president of the section of chemical sciences. And I remember when I proposed his name for Professor R.C. Shah Memorial, he was at that time 44 years. So I asked him, when I looked at his CV, I found him extremely bright. I said, I talked to him on phone, that why don't you wait for a couple of years and we can nominate you for a higher prize at the, say like R.C. Malhotra prize. But he thought a bird in hand is better than two in bushes. So when he insisted, I nominated and there was no question of not getting, he got this award. And he also got a Professor D.P. Chakrabarti 60th birthday anniversary award. He had been associated with the Indian Science Congress, as I say, also he's fellow of the Royal Society of Chemistry. He has supervised large number of PhD students and he has enormous number of research papers, and he is heading many in the committee of the editorial board of Nature, which is a great thing for any Indian to be in the editorial board of Nature, Royal Society of Chemistry, and is serving in many other bodies. At the moment he is occupying a very important position, Dean of Examination of Delhi University. At my personal note, I say that last year when he was appointed as a professor, senior professor in Delhi University, I had the privilege of being in the selection committee. Immediately after the interview, everybody appreciated. There were 3 experts, everybody appreciated the work, and we did not take even a minute to say he's the best candidate and he got selected. Please give him a hand. And on a personal note, I'll take a minute. I am greatly impressed by one of his qualities, which is now vanishing. In ancient time, we had Guru Shishya Parampara. People used to have lot of regards for his Guru. These days we don't see that much. But Diwan showed me a lesson which I also learned at this age. His Guru, D.S. Bhakuni, another Diwan Singh Bhakuni, and as I said, I was also associated with him. He was having 85th or 90th birthday, and he undertook the responsibility of celebrating the birthday of his Guru. Dr. Bhakuni was stationed in Lucknow and he was serving at Delhi University. He wrote to all the persons throughout the country and also abroad, because D.S. Bhakuni did a PhD with Barton, Nobel laureate in Imperial College, London. He collected the family tree of Diwan Singh Bhakuni, right from his forefathers. He collected not only that, family tree of Barton, and then he collected all the contributions from us to write, to publish something. And if I am correct, you can intervene. There was some big family tragedy. His father expired. Now can you listen? His father expired during those days, but he did not stop working on the project to see and do something for the Guru. And then it was celebrated with great show in Lucknow, and so many persons were there. And this is one thing which I learned, that the Guru is great, so much Guru. I don't want to say much, and I request him to deliver this Platinum Jubilee lecture, and time allotted to you, my friend. Thank you.
Thank you so much for your kind introduction, sir. It's an honor to be here to deliver this Platinum Jubilee lecture. First of all, I would like to thank ISCA for selecting me, the sectional committee for recommending my name for this prestigious honor. The front row, my seniors, I cannot say that Professor Singh is my friend already. He's my forefather, like actually, because we grew up by reading his textbook actually on reaction mechanism. This was the book what I studied when I was in masters. Professor Yadav, Professor Tiwari, many more senior colleagues here. Today I am going to talk about the work which we have been doing at University of Delhi for decades or so. Before going to talk about the technical part of my presentation, let me just show the first slide to the students from where the organic chemistry started and from where we are now, and how much development of this branch of chemistry has impacted the human life. If you recall, the first organic compound that was synthesized in the laboratory was urea by Wheeler. We all know that in 1828. And first synthetic drug, that is what we call, is chlorine. It was developed way back in 1869. And after that, you have phenylacetylene derivatives. Then aspirin came in the early 1990s. This year, 2022, about 37 drugs were approved by FDA. And what was the effect of this? In early 1900, the life expectancy was 90. products isolated from this plant show poor anti-cancer activity. Same plant generated another molecule, wherein these two molecules are hooked up by a covalent bond. This is called vincristine, another molecule vincristine. Vincristine and vincristine approved by FDA in 1960. This is the structure of chloroquine, and these are two other molecules called cycloguanine and pyrimidine. So what we did, we took this portion of chloroquine, what we call 4-aminoquinolane, and this part of a molecule, and we hooked up by a covalent bond through a linker, made these hybrids. Looks simple, but when we did this was the molecule inside. This is what we call a trizene, aminoquinolane trizene based hybrid. What we proved the concept, this is the IC50 value of chloroquine. The bar height is higher. But if you look at some of the molecules have IC50 better than chloroquine. So it means the concept what we thought it worked. But we started in 2008 because in the university system we have so many problems. Your characterizing get delayed, biological execution get delayed. So by that time, two papers came in 2009, one from CBRI and one from Argentina, on this particular concept. Yes, hybrid molecules and anti-malarials can work. Luckily, our compounds were totally different than what was published. We could publish two papers out of it. The next goal was, in medicinal chemistry, what we say, that change the molecule at one change at a time. So you develop a structural activity relationship studies. So once you have this kind of molecule, the first idea you can think of as a medicinal chemist to remove one of the nitrogen, keep everything safe, study the biological activity and see what we get. So the next one was the pyrimidine based hybrid. In this case, you get two regioisomers. One was major, another was minor. We converted the major one, made the final compound, something like this. And when in vitro activity of these two regioisomers was carried out, what happens if you look at this molecule? Look at the attachment here. If this is attached at this position, in this case, this portion is attached with the pyrimidine ring, which is flanked by two nitrogen. These are two regioisomers. But if you look at anti-malarial activity of those molecules, it is almost identical. It means the point of attachment of aminoquinolane with pyrimidine has no effect on biological activity of those molecules. That was a good news. So we converted this using the carbocyclic amine. The activity was improved tenfold. From this, when we went to N-methylpiperazine and N-methylpiperazine, the activity was at 7 nanomolar. So whenever you have a nanomolar range activity, people will ask you what about the in vivo efficacy of those compounds. We did in vivo activity of those compounds. What we found that it cures 80% of the mice infected with plasmodium falciparum infection. So in vitro good, in vivo good. Then we carried out a structure lead optimization program. This was the molecule which I discussed before. Different changes at different positions. So we started from this 2.45 micromolar to 7 nanomolar, and these were the other molecules what we see. So I will just give you one example among these designs. What we did actually in first case, what I showed you, 4-aminoquinolane and pyrimidine, you have a flexible CS2 linker. So we thought these two pharmacophores are hooked up by a rigid pharmacophore like piperazine. When you say piperazine is a cheerful formation, cyclohexane chemistry will come in the future. So when we did this, and we made these two set of compounds, in the other set, we kept the flexibility here to understand whether this has any role in antimalarial activity or not. Results were very interesting. So this was the original molecule which I showed in the previous slide, 7 nanomolar active. When you hook up these two pharmacophores from the rigid linker called piperazine, activity drops. But when you introduce the flexibility and keep piperazine to this side, the activity improves, and this is 53 times better than chloroquine in vitro. In vivo, 100% cure at 33.3 milligram per kg body weight. If you treat for 3 days, after 17 days, all the mice are cured. Just to understand how these molecules are acting, because aminoquinones are known to inhibit the heme actually, hemogen. So this is the UV studies of hemojoin. So it stops the hemojoin formation. So this is the absorption maximum of heme, and when we titrate this heme with our compound, the absorption maxima drops. It means what? Our molecule interacts with the heme, and that is the mode of action of those molecules. This was a beautiful design actually. Why? Because if you look at this molecule, this is called amodiazine. And I showed you before how difficult it is to bring a molecule in the market. Right? It takes a lot of effort, a lot of money. This molecule was licensed as antimalarial drug in 1960s, considered to be better than chloroquine, but after a few years, it was to be withdrawn. It was withdrawn from the market because it has a liver toxicity. Why this molecule shows liver toxicity? Because in presence of P440 cytochrome enzyme, this forms this kind of quinone system. And this quinone system is toxic to the liver system. And if you look at the paracetamol structure and its metabolism in liver, it's the same one. And this information was known in the literature when this molecule was discovered. People did not pay attention to that. As a result, this molecule was discovered. Now, as a medicinal chemist, if you want to reduce the toxicity of this molecule, then you do two things. One, find the position of phenolic OH and this end-diethyl. Just find. If you do that, you won't get this metabolism. But whether the activity will be there or not, that we don't know. Another one is, you substitute this OH by similar size of atom like fluorine. It was done through non-toxic and equal activity of this molecule. What we designed here, we designed this. So we kept this portion of the molecule, fluoroamodiazine, and this portion was taken from our previous studies, what I showed. So these kind of hybrids were made. So I'm not going to talk about the synthesis here. We got two regioisomers. What is the difference between the amodiazine and other molecule? OH is substituted by fluorine, and diethyl group is substituted by pyrimidine. What's the activity? It is 100% cure. And if you substitute this phenyl ring by cyclohexyldiamine, the activity was 2.2 nanomolar. I'm not going to talk about that one. But at this particular point of time, we published this paper in 2012, and I received an email on Friday morning from Professor Kim from Harvard Medical School. He said that he is interested to screen our compound for Parkinson model. And I knew one paper wherein the chloroquine's Parkinson potential was published in 1987 in Nature. That paper concludes chloroquine or similar drugs could be used to retard the progression of Parkinson's disease in human patients. But there was no follow-up on this. So we started working with him. What he wrote to us, he screened 960 FDA approved drugs, got 3 hits, and structure of all these 3 compounds are listed here. All have 4 aminoquinone formation. Our compounds is same, but hybrid of different formations. So that prompted us to accept the collaborative offers from Professor Kim, and we started working on it. Let me just give you a brief introduction about the Parkinson. As we know, it is a neurodegenerative disorder. There is no drug available for the treatment of Parkinson till date. L-DOPA is given just to minimize or stop the progression of the disease. But if the disease is at the advanced stage, L-DOPA also does not work. Why it happens? It happens because of the selective death of dopamine neurons, and those dopamine neurons actually synthesizes dopamine, and dopamine is very crucial for the functioning of the brain. So when the dopamine neurons takes place, the dopamine function decreases, and you see all the problems of symptoms of Parkinson. So it means what? It means if you want to work on developing a molecule that can be useful for Parkinson treatment, you need to stop the death of dopamine neurons, number one. Number two, there is another protein called alpha-synuclein. Misfolding of the Parkinson patient, it has been shown that the misfolding of this protein takes place, which is called Lewy bodies, and that is the biggest problem. So if you can stop the aggregation of this, or this one, you can develop a molecule for Parkinson. To show you that symmetrically, this is the substantia nigra part of the brain. This is the portion where the dopamine plays a role. So in the normal healthy brain, you have a dopamine neuron, dopamine is synthesized, and you have a normal function. But in case of the Parkinson disease model, the dopamine, the cells dies, you don't have a dopamine here, you have a movement disorder. You have the protein, misfolding takes like this, you have a Parkinson disease. So all you need to do to stop these two processes so that you can have a molecule that can be used for Parkinson treatment. So we started working on this actually way back in 2012. In the initial stage, we set about 120 compounds. Of these two, one is trizine based, another is pyrimidine based. All trizine based compounds are totally inactive. But these compounds, 4 of the compounds from initial studies, we found that it activates the neuron enzyme up to some extent. So we started working on it, and we developed about 650 or 700 new molecules, multiple step synthesis, and we found about 50 molecules. The chloroquine's EC50 value is 50 micromolar. Some of the compounds which we developed, the EC50 value is about 1 nanomolar or 0.5 nanomolar. Highly active molecule. But to develop a clear-cut SAR in this system is a really, really tough task. In medicinal chemistry, most of the time what happens, you make a systematic change and you could draw conclusions. But in this case, that was not possible because people felt that neuron is an orphan receptor. But now it has been established that it has a binding pocket, but the binding pocket is very tight. So you need a molecule of a proper size which can fit in that and activates the neuron enzyme. So while doing this, actually, we published a paper in Royal Society RSC Advances in 2015 on antimalarial activity of these molecules. So this was the molecule which we published, and I wanted to do the detailed biological studies of this molecule. And Satya Pawan was working on it. And I asked, make this molecule 100 milligram quantity so that we can do the detailed biological study on it. During this synthesis, he reported that he got another polar compound in 100 milligram scale, 8 milligram of that polar compound, he gave it to me. This is what isolated, which most of the time the PhD student misses out. The structure of that compound was this. And this compound turned out to be the best one in Parkinson model with 0.5 nanomolar activity. Then we improved the synthesis of this molecule, made in a larger quantity, submitted for bioscreening. Then there are many other molecules for synthesize. We have about 15 such compounds with us now, which has very low nanomolar activity. Just to remind you, what we are looking here, you are looking here a molecule that can activate the neuronal receptor so that the depth of dopamine neurons can be increased, or a molecule which can stop the aggregation of alpha synapses. These are the two things what we are looking for. The bioassay, this is the luciferase assay using neuron binding domain of a molecule, and the green one is the IC50 value for chloroquine, and red one is our molecule. The consumption is 100 micromolar to 20 micromolar. So our compound was 5 times better than chloroquine, and this assay is the full length neuron enzyme, same result were obtained. Then you have a vehicle, this is the dopamine neurons. You have a red dots and the green dots. Okay? And when this is treated with NPPP, NPPP is a compound that causes the Parkinson. So this is the typical model, Parkinson model. You take that molecule, inject to the mice, and the Parkinson symptoms will increase. Why? Because the death of dopamine neuron takes place. Another one is LPS, also does the same thing. You can see in this slide the red dots decreases now. When this is treated with chloroquine, the red dots regains little bit, but when you treat with our compound, the red dots increases much more. 62.7% of the dopamine neuron were rescued. They survived it. So it means it cures the death or stops the death of dopamine neuron. As a result, you will have more consumption of dopamine, so the Parkinson problem will be resolved. So this is the substantia nigra part of the mice. This is the vehicle. When treated with this molecule which causes the Parkinson, you can see the brain structure. But when it is treated with our compound, the last one, it looks almost identical. So it means the molecule what we are looking for does what we wanted. The next one was it stops the aggregation of alpha-spin protein in substantia nigra. This is another mechanism. So this is the vehicle again and treated with our compound. If you look at this structure and this structure, both looks almost identical. It completely stops the aggregation of this protein which is responsible for Parkinson's. The third important thing, it induces autophagy also. The students, do you know autophagy? Sunat? Autophagy means when your system is under stress. Okay? The healthy cells eats the cellular garbage. Okay? So the cells which are dead or causes disease can be killed by your own system. 2015 Nobel Prize was given on that. That concept is called autophagy. So the first one, this one. So this is the normal vehicle, and under the starvation condition, when this molecule was treated with this media, it starts to autophagy. What it does mean? Yellow dot and red dot increases. It means it is inducing the autophagy. When this is treated with this molecule, the red dot depletes. It means autophagy stops. Now this is treated with chloroquine. Little bit red dot increases. When treated with our compound, it shows 63.9% at autophagy. So this molecule, one molecule, is activating neuron receptor responsible for the death of dopamine neuron. So dopamine neurons death stops. Number two, it stops aggregation of alpha-spin protein and it induces the autophagy. So this is what it does. So from this, we could rescue this mice from the Parkinson disease. Parkinson was induced, treated by a molecule, completely finite there. It has a good pharmacokinetic properties, and these are the students who have contributed to this work. So in 2012 we started, we got 4 papers, no papers here. So you can understand how much patience is required to work in these kind of diseases. projects. So ultimately is this, because these three molecules are listed on the website of this company now. It's in the Boston-based pharmaceutical industry. They have transferred the technology. Now they are taking up these molecules further to develop as a drug for the treatment of Parkinson's disease. In between, because as I said, it takes a lot of time, a lot of patience in medicinal chemistry. You make at least 30-40 compounds, get them screened, that takes time, and the data comes, it's not interesting. So you can't even publish it. Right? In between, frustrated, I said, okay, we start with a bit catalysis work also. So we continued in this area also. A lot of papers have been cited well and got the cover page also and highlighted by Sinfel. This was possible because of generous funding from CSR, DST, UGC, DST Pulse, MJ Fox Foundation, funded or grant on Parkinson and Institute of Immunity, Delhi University. All malaria work was collaborated with Dr. Maharaj Nagpur because nobody wanted to collaborate with us when we started in 2003. Because when we were young, people don't take much of the given importance. So everybody whomsoever I contacted at that time showed inability. But she has done this, and Professor Kim from Maclean in Harvard Medical School, they did all Parkinson related work. And what I presented today, it is because of the dedicated and hard work of all these bright students. 26 students have got the PhD, they are well settled. And Mohit Tripathi left my group about 6 years back actually. Still, whenever I call him, he comes forward to work with me on this Parkinson related work. And this is the current batch of students. And thank you very much for your patience here.
Very illuminating talk. What I gather, that focus has been on the development of anti-Parkinson vaccines, and second is on anti-malarials. Both are difficult problems. Of course, I will say for the development of anti-Parkinson is a focus throughout the world. Everybody is working on, and there has not been any substantial progress in this direction. But from our context, from Indian context, we feel developing anti-malarial scaffold is most important. I am saying it because of two reasons that say malaria, there is not much research on malaria in advanced countries because they consider it a neglected disease. It is a disease of our country, not in Europe or America. So it is us, Indian scientists, who have to get drug for malaria. And your efforts are commendable. And second loud message that you have given, that you have to look to nature for getting treatment for malaria. You know, the first drug that came, quinine, 100 years before, from Sincola. The second drug that came from Chinese, artemisinin, again from Artemisia animal. And the person, it is interesting that the person who discovered artemisinin, the lady Yu was not a professor, was not a PhD, was not teaching in university, was not a scientist. She was a pharmacist. And just by taking the help of the Chinese system of medicine, because only two systems, traditional systems of medicine are important in the world, Chinese and Indian. She took help of the Chinese tradition plants, tested them on herself, others, and came out with artemisinin. And my young friends, she is one of the very few persons who got Nobel Prize in 2015. Few persons means the person who was not having any degree, PhD or professorship, yet she got Nobel Prize of 2015. So I suggest my young friend, thank you very much, and I suggest you focus more on neglected disease so that you may help the country. Thank you.
Professor S.P. Singh sir to felicitate Professor Diwan Rawat sir with a memento as a token of love, appreciation and gratitude. Thank you, sir. May I request Deepika Gidkar ma'am to please occupy the seat on the dais. To chair the session.
It's a great honor for me to chair this session. I never thought that I'd be doing so. When Professor Yadav, whom we know of, at least the younger people like me, I consider myself young guy, still I do. So he's a kind of role model for all of us. What he has achieved professionally is commendable. He is one of the highly accomplished engineering scientists in India, and he is the Padma Shri, got the Padma Shri Award. And he is the fellow of all three National Science Academies. He is the fellow of Third World Academy of Sciences. He was the Vice Chancellor of Institute of Chemical Technology in Mumbai, and it's a commendable job. He took that institute to a new height, and I think he established two more, one in Bhubaneswar, another in Jala. So two of those were instituted by him. He is a J.C. Foods fellow, and currently he is the president of Indian Chemical Society. Without taking much time, I humbly request Mr. Yadav, please deliver your talk. Thank you.
Because I have a flight to catch, and they said 4 o'clock, so I believed at 4 o'clock it will start, and it was not going to start, so I'm very sorry for that. So the young girl can wait for some time. Ladies and gentlemen, you have heard this buzzword in all newspapers. You read it, whether it is vernacular or English or whatever, the net zero goal. And it's just, is it a political term? Or is it science, engineering, what it is? And how we can achieve sustainability by using green hydrogen technologies. I've been working in this area a number of years. I started a program in green chemistry and engineering in ICT for chemical engineering students in 1998. And it went into separate program with them. Then CO2 refineries and biomass valorization. Will you please close this line? All of you are aware of Paris Agreement of 2015. 195 nations signed this agreement. our Prakash Javadekar Honorable Minister were there, except the United States of America. Except the United States of America. And what did they say? That we will have temperature rise, global temperature rise, not going beyond 2 degrees C during this century, and particularly less than 1.5 degrees Celsius. So this is a lot of common sense. I want to tell you something. And so there was a issue. Donald Trump did not agree with it. But Joe Biden, who was contesting as the president, committed himself to the net zero goal. And he said we have something. And immediately after taking over as the president, he organized this leaders summit last almost 2 years. Our honorable Prime Minister was present there, and he recommitted himself, United States as a nation. Now then, in 2021, this COP26, Government of India committed to net zero by 2070. What is meant by net zero? Carbon emissions will be neutral. That is net zero, net carbon zero. So my question, this is a political decision by the government, we respect it, but my personal belief, why not 2050? The entire world is talking about 2050. Is our science and technology capable of not doing it? Indeed, we can do it. So I'm going to show you how we can do it, okay? Next, then this year also, last year in Egypt, we had this COP27. Once again, India wanted something on the coal-based technology, some concession, which they did not agree. So I'm going to talk about a little bit of this. There was a movement in South India, those from South India, you know that at Kudam. A lot of people were opposing the nuclear energy. There were demonstrations and demonstrations. Then there were NGOs who were saying that we should stop our nuclear power and we should not have coal-based power. And they were giving examples of Germany where both of these things were stopped. Now, after the Ukraine-Russia war, what happened? The same Germany which was giving a big bhashan to us and our NGOs were telling we should not do it, they restarted their plants. They restarted their nuclear power plant, they restarted their coal-based power plants. Perhaps you don't know, the uranium which is purchased by United States is still purchased from Russia. But then they tell us the third world countries which are highly populous like India, don't use this technology, which is nonsense. Alright, so you see that this data which are available from 1750 to 2018, USA, how many billions of tons of carbon dioxide are emitted? 397. India was number 7 in that. However, look on this side. Now, the megatons of CO2 per year, you see, we are number 4. That means because we are highly populous country, now almost 1.4 billion plus, we are emitting a lot of carbon dioxide. So how do you talk about carbon neutral technologies? So let us look at this. So I have a prophecy. Mark my words. India will be a leading exporter of green energy by 2030 and your import bill will be reduced. This is what the government wants to do. This is what the planners want to do. Then green hydrogen will be less than $1. I have been saying this since 2015. I can produce hydrogen less than $1. And my research has been supported by ONGC Energy Center. But they were dilly-dallying. And now everybody says, do it fast, do it fast. Do it fast. So I've been in fact given a time limit that I must do this, develop this technology, although I've got so many patents across the world. And they say it is India's century and India will be the third largest economy. Honorable Piyush Goyal said we will have $30 trillion economy in the next 30 years. It cannot be achieved without manufacturing. Service sector is not going to give that. Right now we are only 3.2 trillion. Okay, USA is 23 trillion. China is 14 trillion. How do we achieve that without manufacturing? And that has to be indigenous manufacturing. And if that happens, if the net zero goal is achieved by 2050, will the world be peaceful? The United Nations 17 goals. We said zero hunger, no poverty, all these numbers, 1 and 2 goals. Number 9 is industry, infrastructure, and innovation. All these things will fall in place. What is the reason for this? Greenhouse gases and effect, because ours is a carbon-based economy. So we have this carbon coming from oil and natural gas or coal, and we have many different technologies. Now people are talking about crude to chemicals. Most of the refineries in the world, they have fuel-based technology. Only limited part goes for chemicals. And by 2050 we will not have any crude oil. So what are you going to do with our refineries? Okay. So there we have this renewable carbon, biomass. Many of these things, we won't go into the details of this, but this we argued in one of our papers, which is highly downloaded paper. Many bureaucrats in USA, Canada, they contacted me. We gave a new thought to this energy scenario. So I'm very glad. What thought we gave? So energy and environment and the climate change, they are all linked. Okay. India is a leader in solar energy. We'll talk about that also. But remember, since you are chemistry students, you should know the value of chemistry and chemical industry. Many NGOs say we will not have chemical industry. In fact, one of the retired judges wanted to argue with me on television. I said, you are a judge. I can't argue with you on the law. And you should not argue with me on chemistry or science, because that is not your domain. And he said we should close all chemical industry. So I asked him, did you eat any medicine in the morning? What is there on your body? Everything is a part of chemical industry. It is a produce of chemical industry, including your mobile phone. There are 20 different types of chemicals, including gallium arsenide, silicon and whatnot. We have to produce them, the polymers and whatnot. So anything which you touch here is a produce of chemical and allied industry. And if anybody can show any 3 products which are not produced by using chemicals, they will get a prize of 100 million pounds. Which is waiting since 2008. Some gentleman announced it. So I was telling my friends in industry, because I'm associated with CII, FICCI and ICC, I said, let us announce a prize of 100 crore. Even I said, I will announce it. I don't have that money with me, but I know nobody will win it. I'm sure about it. So I said, at the most, what is going to happen in future, Queen will be replaced by Charles. That is what is going to happen. But the concept is very important. You cannot do without chemicals. Long time ago, in 2001, I was the president of Indian Institute of Chemical Engineers, and I had a meeting like this in Bhubaneswar, and one of the bureaucrats said, we are going to convert Bhubaneswar, now where I started a campus now, that it will be a cyber issue, no chemical industry. So I was president, okay? So I had to say, I said, my dear sir, I'm going to educate you now. If you think that chemicals are not required, you have to go home without anything on you. You'll have to hide your modesty. So would you like to do that? You can say that we don't want pollution. Absolutely right. We don't want any polluting technology, and everything what you talk about is also creation of chemistry, whether it's a 3-way catalyst and whatnot. So jokes apart, we produce a lot of waste. You must have seen all this waste, right? So I'm going to talk about waste, how to stop this waste and how to convert that into chemicals and materials. So the sustainable development goals must be socially acceptable, environment friendly and economic. That is what we call sustainable development. And in this you will see that the goal 1, goal 9, these are very important. So one of the things we can normally talk about is circular economy. What is meant by circular economy? Okay, and the recycle engineering. So look at this. We have a lot of things. The physical, chemical and biological processes. And these are all rate processes. And I'm a chemical engineer, remember that. So everything we talk about rate processes, we say process intensification. We increase the rate of reaction, reduce waste byproduct, okay, and convert. So atom economy, green chemistry you talked about. Green chemistry, atom economy. How many atoms I can incorporate in the final product? If there is no atom there in the final product, why use it? So yes, so material recycling, sustainability, and we have to create a zero waste society. So I'm going to tell you that also. So since we are talking about solar energy, wind energy and all, there will be a lot of waste because these panels will be, one day they will not be useful. How do you recycle this material? If we do not do that, we will require 3 Earths. Now this number 3 has gone to 5. We are producing so much of waste. What do we do with that? So I will tell you something else also. one of the things is we are talking about batteries, right? The e-vehicles, and by 2030, Honorable Gadkari ji says, I've shared that with him, that we should have e-vehicles. But if that e-vehicles, electricity is coming from coal, actually your battery is coal powered. So your vehicle is coal powered, not battery. Electricity is required. So electricity should be generated by renewable non-carbon resources, non-carbon. Otherwise our carbon dioxide emissions. So the battery market, you see that, how much of this, the recycling battery is a very big industry. Because when this battery goes down to 80%, it is useless. So can it be used for something else? So that is called second life for energy re-engineering. So we can see that. So there is a recycling of this plastic, lead, electrolytes, you name it. So recycling, that is a very big business, and you should do research in that area. You should do research in that area. So whatever type of, whether it is, and we don't have lithium. We are talking about lithium batteries. Okay? We don't have lithium. So what do we do with that? So remember this, see, China in the circular energy storage, China dominates. They have something like $45 billion 2030. Okay, so recycling, you see 70% of the electrical vehicle batteries from e-scooter to buses by 10 years time could be remanufactured into other vehicles. So this is a very big interesting area. Can we reuse these materials? Battery is one of the things. Okay, so battery recyclability is highly important. China has a growth rate of 9% in that business. USA has about 3%. In India, we are picking up. We are picking up. I'm part of this MSME committee, and I'm telling you that India it is picking up, but not so much. So you see that this is CAGR of 8%. And it has, incremental growth something like 62.7, $77 million. So battery recycling market was 8.74 billion in this, and so it is very important that use of recovered metal from recycled battery production can help in reducing CO2 emissions, because ultimately we want net zero. The CO2 emissions have to be replaced by any and all means. So what we do, so look at this is very interesting slide. By 2050, we will have 73% of our energy coming from renewable resources. India is a global leader, you know that it is solar energy. We are talking about something like 900 gigawatt of solar energy, and then we'll be an exporter. That is our government's aim, and that is very good aim. So how do we do that? 450 gigawatt solar power requires $500 billion investment. It's a lot of investment. You remember the economics of that. So here, our energy is 73%. In that, what will be? Solar energy, wind energy, hydro, nuclear. And in this particular, 49,000 terawatt hour energy, 24% will come from hydrogen. It will be both green hydrogen and blue hydrogen. And we should promote green hydrogen. So this is why I'm saying that some of the interesting facets. The so-called biorefinery is not a new concept. Green chemistry is not at all a new concept. I told Paul Anastas, okay, you mentioned Paul Anastas and John Warner, both of them are my friend. I said, you are not the originator. This was already done by Solvay process. Solvay process is the first example of green chemistry as well as chemical engineering. Okay, when synthetic ammonia was also not there, but they did it. So these oileries, this was the first biorefinery. What were they doing? They were capturing these wells, take the blubber out of it, and convert that into oil for illumination and soap making. That was the business of that time. So what happened? Over the period of time, the well population went down. And this is the time when we have this carbon-based economy. And we have to thank. So avalanche of this coal and crude oil as providers of energy, chemicals. So we have this example of Edwin Drake in 1859. He placed the first well in Rashtrasant in Pennsylvania, and the rest is the history. Initially, there was no use for this gasoline or petrol. That was literally dumped into lakes, seas, and whatnot. Only in 1906, Henry Ford introduced the first vehicle, and we started using petrol. And all the technologies were developed thereafter. Okay, all the technology. Very interestingly, what happened? You see that in 1940s, we introduced this clay-based catalyst for cracking. Zeolites and all that mobile in 1962 and thereafter. But this is what happened that time. Very interestingly, the Allied forces, which included Russia, they won the war against Nazi Germany because they had superior quality of aviation fuel. They had better catalytic technology. So catalysis has played a very important role in avoiding wars or winning wars. And that time there was a Russian general, Vladimir Ifatiev. Vladimir Ifatiev, was a great scientist. All this platforming, cat cracking, whatnot, he has developed it. At the age of 60, he retired from the military, went to the Germany by train, told his wife, this is the last time you're seeing Russia. He never knew any word of English. He only knew German and Russian. The EOP, perhaps you have heard the name EOP. EOP is a great company which developed so many petroleum refining technologies. They hired him, secret dealing. But he had a girlfriend. They said, we'll take your girlfriend also. That's a fact. It looks amazing. But they took her also, they paid her because he was a great scientist. He was nominated for Nobel Prize many times, but because he was a Russian general, he was never given. Like Mahatma Gandhi never got Nobel Prize. So sometimes history is very bad. Sometimes history is so the person who deserves doesn't get it. Yasser Arafat got it, but Mahatma Gandhi never got it. You know that. I will not go into the politics of it, but this is the fact of the matter that, refinery technologies are very complex. So this is where now we are talking about hydrogen economy and green hydrogen. So the only way to stop this, the denudation of forests, reducing the temperature of the globe is using green hydrogen. Since I've been involved here and got several patents in the area, I want to tell you something about it. So I am calling a new trinity. Brahma, Vishnu, Mahesh, right? We know the Hindus, and the Christians call Father, Son, and Holy Spirit, right? They are borrowed from Hindus, I can tell you that also. I can give another lecture. On that. But this true trinity, solar energy, wind energy, and hydrogen energy. And hydrogen takes the topmost position here because hydrogen can give you both energy, fuel, materials. Wind and solar will give you energy. So this is the case. So I developed this technology called ICT, Institute of Chemical Technology, ONGC Energy Center. We got several patents. We are building a plant in Goa. Another will build in Andhra. So I have been told by the government I must do it within the next 3 years because mine is not based on electrolysis. Prime Minister yesterday mentioned about electrolysis. Why the government is interested? Because the electrolysis is not efficient as compared to this thermochemical process which we have done. So what we have done, so I am telling you a very interesting story. The Ministry of Power made this notification on hydrogen policy. And now today only I read another thing, green hydrogen mission. Government has just announced today. Right. So what is that? That means water and air will be our feedstock. We take hydrogen from water, that is green hydrogen. We take oxygen also, which is required, and nitrogen from air, pressure swing adsorption. Chemical engineers would know that, and that will be and what is government objective? 50% of the energy requirement be made by green hydrogen and green ammonia. So why green ammonia? So look at the synthesis of green ammonia. The Haber-Bosch process is no. Okay, so what we are going to do here, we take solar energy, wind energy, hydro energy, split water, either water electrolysis or thermochemical, which we are doing and we got all these patents. Get hydrogen, from air pressure swing adsorption, take nitrogen, you make green ammonia. And this green ammonia directly can be used for many things. Ammonia gas turbine, ammonia boiler, ammonia fuel cell, or you crack it, okay, catalytically, and use it as a green hydrogen because the other part is nitrogen. So that is the reason why we are talking about green ammonia and green hydrogen. So remember, this consumption by 2050, only green hydrogen and blue hydrogen will be there. Grey hydrogen refineries require hydrogen for 8 processes right now. And what they do that? They make it by the steam reforming of methane, natural gas. So that produces a lot of carbon dioxide, 11 tons per ton of, So this slide I have prepared on the basis of European Union, International Hydrogen Council, Bloomberg energy source. So what it means, in 2020, see, we emitted 35 gigatons of carbon dioxide. And to achieve the goal of 1.5 degrees C, we have to extract this carbon dioxide, come down to 10 gigatons, then only we will be able to achieve 1.5 degrees C. Otherwise, what is going to happen? The problem will be, we will have tremendous quantity of carbon dioxide. In 2050, it will be 8 degrees C. Nobody will survive that. So that means it is technological intervention which is required to bring down the temperature. So hydrogen is the cleanest fuel of this. And I will not go into singing the songs of hydrogen, but I will tell you. It can be used for green power, transportation of 2, 3, 4-wheelers, IC engines directly, synthetic fuels from carbon dioxide. So carbon dioxide can be converted into a variety of things. So we have many patents in that. We have done that. Upgrading of bio-oil, then hydrogenation of biomass, green ammonia, fertilizers, green metals, steel industry is highly producing. I can make green steel, green nickel, chemicals and allied industry, waste plastic chemical recycling. I'm going to talk a little bit about that. And heat and power. So the hydrogen economy, to be a reality, I've been writing articles, editorials and whatnot. To be a reality, we must have a green hydrogen policy. Government of India must adopt hydrogen economy. And why? I will tell you many things about that. So one of the things is, for example, so we would know about green hydrogen, blue hydrogen, grey hydrogen. Green hydrogen is electrolysis water or the so-called thermochemical process which we followed. Blue hydrogen is steam reforming of biomass, any carbon source, but the carbon dioxide has to be captured at least 90%. And grey hydrogen where you don't capture the carbon dioxide, which is right now being done. Okay? So, or you can have methane pyrolysis. You make hydrogen and the solid carbon, which could be carbon nanotubes and whatnot. So, very importantly, why green hydrogen? Because water splitting, both hydrogenation and oxidation of waste biomass and clean biomass, purposely grown biomass into chemicals and materials. So I will tell you a little bit about that. See what our farmers are doing. They are burning this. This is the waste of national energy and creates a lot of pollution. If you are staying in Delhi and around, you know what it happens. See, the air quality is very poor. 300 million metric tons every year is burned by farmers, which can be converted into variety of this. And very interestingly, all of these reactions are based on hydrogenation, hydrogenolysis and oxidation. And that hydrogen comes from water. The green water, oxygen also coming from that, that means it is the best strategy to use. So I mentioned before, hydrogen, coming from water and nitrogen coming from air. So you see how many, all these reactions we have studied in our laboratory. I won't go into the details, but any carbon source is good enough for converting into chemicals and materials. So the farmers can also benefit by hydrogenation of waste biomass instead of burning it. Okay, so if you look at new cooperative society, now your cooperative society is for sugar factory, right? We can do that other way around. So no matter what carbon dioxide you have, whether it is from biomass or crude oil, ultimately it is carbon dioxide valorization. So carbon dioxide is not a liability, but it can be an asset. So I must have technology to convert carbon dioxide. So carbon dioxide refineries is a concept based on that. So I can produce a variety of chemicals like methanol. Niti Aayog is talking about methanol economy. Methanol can be blended. Then we have dimethyl ether. All these things, dimethyl ether is a very good substitute for diesel. It is a substitute for LPG, and same infrastructure can be used. Unfortunately, our country doesn't have dimethyl ether policy, although it was thought way back in 1995. Indian Oil had thought about it, but they did not implement that policy. So now I wrote to the government, I said, you must have a policy. Why? Because I will tell you in the secret. So CO2 based chemicals, and so many times people talk about salicylic acid, that is a pillu, it is very small quantity. What we require is a huge quantity, huge usage. So carbon dioxide can be converted into that. So we talk about that. So methanol economy. So we have written a very big articles on that in green chemistry and whatnot. So the next question is, should biomass, bioethanol be burned as a fuel? That is $1 billion question. So we said it should not be. Now it is against the government policy, right? So why I said that, I will tell you. The reason is very simple. Look at the hydrogen usage in oil refinery and biorefinery. So this is the so-called, gray hydrogen, which is used 8 different processes. These are the refinery processes, cracking, hydrocracking. These all require hydrogen. And right now they take this hydrogen from steam reforming of, gas. At the same time, we require it here. So we can compare petroleum refinery versus biorefinery. In one refinery, you go on functionalizing the material. You take benzene, toluene, xylene, butane, these are your feedstock, and you convert them into something else. So it is functionalization. Here exactly opposite we are doing. We are doing defunctionalization because we need the molecules for our feedstock, the refineries. So we do that. So we did some calculation, very interesting, which we have given this in this paper. So if you take 1 kg of, this is the current refinery, that time their production was 97.4 million barrels per day. We get 32 megajoules of fuels plus 0.2 kg of chemicals. This is the typical structure of a refinery. However, if I take 1 kg of biomass, I get only 6 megajoules of fuel, but I can get, instead of that, I can get 0.8 kg of chemicals, valuable chemicals. So see, based on that 97.4, which is 250 barrels per second, 100% biomass converted into fuels only 10 barrels per second, but this is only 320 barrels. So you imagine, that means converting biomass into chemicals is a better idea from environmental problem than, what is happening? You came here and shouted. No, I'm just kidding, okay. This, what is happening here? There is an error. Okay. I hope it is there. So we can, so now see, I mentioned that bioethanol should not be blended. Government can continue to blend till they find a solution. But what is the real objective? See, bioethanol I can convert into variety of chemicals. I go to ethylene, dehydration. So earlier they used to do it, but we started getting ethylene from refineries. So we forgot about it. So all these chemicals can value add by ethanol. So carbon should not be burnt as a fuel. That is my message. If you want to achieve the net zero goal, carbon should not be used as a fuel. Use anything else, non-carbon, including nuclear energy. And nuclear energy is the safest form of energy. Many people don't know that. They think nuclear energy means bomb. So we have to educate them. So if you want to achieve the net zero goal, carbon should be used as a source of chemicals and materials. So look at the kind of things. So we have in biomass cellulose, hemicellulose, lignin. And most of the time the lignin is burnt. They should not do that. So you have xylose, arabinose and all these things. C5 and C6 sugars. In fact, I used to tell jokingly to my friends, you don't have to grow sugar cane. You take any biomass and if once have C5, C6 sugars, I can convert it into all sorts of sugars. Okay, so lignin-based products, you see variety of things, starting with syngas. Once you have syngas, you get Fischer-Tropsch synthesis, catalytic processes, convert into anything. Hydrocarbons, including benzene, toluene, xylene, I can produce from this material. Okay, so you can produce a rich catalog from C5, C6 sugars, and look at the lignin source. And all these are oxidation or hydrogenolysis. So water splitting to produce hydrogen and oxygen is going to save us. In other words, hydrogen will be the savior of this world. That is my prediction. So we can do that. So there are methods of producing green hydrogen. This is water splitting cycle. I'm not talking about electrolysis now. Sulfur iodine, copper chlorine. So we use copper chlorine. ONGC came to me, they said, can you develop this? I said, yes. So we developed this in 2005 it was. Now I can produce hydrogen at 95 cents a dollar. Ambani's, Adani's, L&T, everybody is talking about this. I have got 6 inquiries. And we are going to give a press release that we can produce. So we can, give this hydrogen to steel industry, we can give to fertilizer industry, we can give to many others, vehicles. Only thing is for hydrogen as a fuel, we have to develop that infrastructure. But fuel cells can be used. So many things are possible with that. So look at this cycle. We have obtained several international patents. We are now building a big pilot plant in Goa, then another in Andhra, and refineries in Indian Oil is approached me, Tata Steel is approached me. So many other companies are interested in our technology because it is non-electrolysis based. So very interesting. So we can see that, so this is the sum and substance of that. We use solar power, we store it in molten salt, we have copper coating, take water, hydrogen, so you can do hydrogenation, you can do carbon dioxide hydrogenation to methane and higher hydrocarbons including petroleum, whatnot, and oxidation of biomass I told you, so I can produce hydrocarbon, methanol, dimethyl, formic acid, fuel cells, and name it. So this is what we have done. This one slide tells what I did last few years. Okay, and the cost of hydrogen also. Electrolyzers, were some $1,100. This has to be brought down to 220, and they say it will be 2040. So there is a concept in America, it is called 111. That means 1 kg hydrogen in $1 in 1 decade. And they have given it as a challenge to everybody. So we have been approached very recently by one Canadian company. They said they will market our technology. So we are going to go ahead with that. As an engineer, I feel greatly honored that somebody from abroad is coming to us to develop that. So the target is very important. So look on, this is the comparison of hydrogen production. This is my last one. Okay? We have no carbon dioxide. Look at the coal base, 19 tons per ton. Natural gas, 11 tons. Natural gas pyrolysis is 0. Steam reforming 0.2 when you have the blue hydrogen. Renewable electricity and the cost, mine is 95 cents. Remember the oxygen, 1 kg of hydrogen gives 8 kg of oxygen, so 18 kg of water are required. And that oxygen cost today, international price is 13 cents per kg. So I can sell that. I can sell that also. Okay, so this is what happens. So we got carbon dioxide to methane and higher carbons. We got several patents, and any CO2 can be used. So we have used CO2 from refineries, but from coal power plants. So ONGC is supporting it. Then DME is an excellent substitute for LPG infrastructure. We have now this group, Godavari Biorefinery, they emit 2,300 tons of CO2 every day. So they have joined hands with me. So we are going to sell this technology together. Then CO2 into methanol, many patents we have got. So we have green methanol, green urea. See, 14 platform chemicals from biomass. We have developed all these chemicals. And Professor Rawat, plastics, people talk a lot about plastic, right? Everywhere we talk about plastic, plastic pollution. People get solution, they are not talking about single-use plastic is bad, is a wrong idea. I tell you why. It's a wrong idea. Because plastic was introduced as a substitute for paper because paper was denuding the forest. So why there is so much of plastic garbage? Because it is free. Because anything which you get free in the marriages, you get all sorts of dishes, right? Malaysian dish, Thai dish, and you want to try it, and you waste it. So you waste 50% of the municipal solid waste contains food. 50, about 12% is this plastic waste. So what I am trying to tell you, that ban is not the solution. So what is the solution? Solution is like a newspaper boy, the housewife never throws newspaper. She will make raddi and sell it. Perhaps get more money. Some of the supplements are worthy of the raddi only. They are not good to be read also. I don't read it, okay, arguments. But what happens? Supposing you had put some tax, okay, some deposit on there, suppose 1 rupee for everything. Another boy comes in the morning, says, Madam, give me your plastic waste and I will return your deposit because it is digitally connected. Maybe he takes 5% commission, your money is back. No waste will be generated, including the polythene bags which are used for, the milk pouches. PET can be used for, milk, that. This is a good thing for milk. But you can use it. And what I can do with the plastic, because it is segregation, 80% of the municipal solid waste cost is on segregation and sorting. So what we can do? Chemical recycling and upcycling. So you see that plastic recycling, what do we do in that? One of the important things in plastic recycling is, you see, hydrolysis, glycolysis, amino acid. A couple of my PhD students are working on that. And any molecule can be handled, chlorine, nitrogen, oxygen. And so what happens? PVC, PET, polyamides, rubber. Hydrogenation is once again coming to our rescue. We can make oil out of it. And the other part, whether it be HCl or ammonia or H2S, that can be absorbed. And these technologies are already known to refining. So we don't reinvent the wheel. Otherwise, because single-use plastic, even the straw you can use. So you have to carry your own stuff, maybe metal stuff. It will pierce your pocket. So why not say that, okay sir, you want a straw? Give 1 rupee, you will return it. So collection at stores for single-use plastic is the best solution. Once I was called by Maharashtra government and they said, sir, want your opinion. I said, don't ban it. Ban is a bad idea. It will, make the police rich, the authorities, because they will raid, and that is not the purpose. Because you are getting it free, you are throwing it. If there was a deposit, you will return it religiously and get your deposit back. Am I right? The thick bag, women never throw. The sari bag, never they throw. They will reuse it. But plastic bag, yes, because it is free. So I am saying nothing is free. So these are all sorts of plastic waste, catalytic technologies, including glycolysis to hydrogenation to oxidation. All that can be used and reused. And I said in the beginning, recycle engineering, whether it is chemical upcycling, downcycling, all those things. So for example, PET is waste. We can convert variety of these things in different chemicals. And steel, we produced last year 106.5 tons. This year it will be 102 million tons. 2030, 300 million tons. And 1 ton of steel produces 2.3 tons of carbon dioxide. So green steel means what? You eliminate the current methodology, use green hydrogen there. You will have 0 carbon dioxide. That is the solution, not what we are doing right now. So the policies have to be changed. So in this, I have looked at the technology maturity, and I will tell you, I compared my process with published literature and the investment cost. Okay, so you will see that mine is 20 years. I produce at 0.95 cents. Many technologies are talking about $5, $10, $8, depending on the production capacity. I say 100 tons per day, I can produce less than $1. If you reduce it, the cost will go up because it is highly capital intensive industry. Okay, so that is what happens. So cost of electrolytic hydrogen in the future, it has to be less than $2. And I said 111. Remember that number. 111. In cricket, it is called Nelson, right? So you know that, right? So by 2030, hydrogen economy will be a reality across the world, and hydrogen will save us. We will produce something like 530 million tons of hydrogen. Okay? And look at the different uses by 2050. When society accepts this, hydrogen economy will be a reality. And Honorable Nitin Gadkari is driving a hydrogen car. You know that. He is the first person in the country. So he is a great supporter of this idea. And so hydrogen safety. People think hydrogen means hydrogen bomb. It is deuterium, not hydrogen. Hydrogen is safer than your petrol and diesel. All tests have shown that. But we think hydrogen is more dangerous. Hydrogen is 14 times lighter than air. And we have got so beautiful sensor technology, we can detect any hydrogen disease. So hydrogen is safer than fossil fuels. This is the message. Okay? So hydrogen is that conventional fluids. And finally, each work has to pass through 3 stages. Ridicule, opposition, and then accepting. That is what Swami Vivekananda said. Hydrogen, same thing is going to happen. People will accept that as a society. And so then we talk about green economy, bioeconomy, circular economy. These are buzzwords we can. And so we can literally have a zero waste society. If we want to survive on this planet Earth, we have to have the objective of zero waste. And it has to be done only through chemistry, chemical sciences, and engineering. So if you are a chemist or a chemical engineer, I used to say this joke, those then I am telling you this joke. If there was a queue here, with a varmala, a bride coming to choose the groom, and all sorts of guys are standing there. She will not choose a chemist or chemical engineer because they think that they are dirty guys. Why choose them? We would have remained unmarried, I'm telling you. That is the perception of society. So we have to change the perception of society. Without chemical sciences, you cannot survive. Life, chemistry is life, not physics, okay? So ultimately, green hydrogen will be the savior of the world. CO2 should not be a liability but an asset to convert. So my technology is one of the technologies. Government is encouraging me. So I must thank a large number of my students Because of my work, I became the US Fellow of National Academy of Inventors. I'm the only second Indian to be honored in its entire history, and I'm among the, and this on 8th February last year, I'm one among the 21 Indians in the history of 160 years of US Academy. And when I went to receive that honor on October 2nd, I felt out of blue. So many big people, scientists and Nobel laureates. So it's a great honor. It's a honor for the country. And I'm humbled because all my students who are there senior, they were working day and night, like Professor Diwan. It is students. The professor take all their credit. They go in Indian Science Congress, give lectures, make people laugh. But the actual work is done by the students. So we must, salute to our students for doing this. And ladies and gentlemen, I interrupted the young lady. Now you can take some inspiration from me, give a good talk. And I want to thank you all once again for inviting me for this wonderful meeting. And I've been staying, and one number you should remember, I produced 107 PhDs. Not even one of them without fellowship. I have not taken any government money for last 22 years. It is all money generated through master. Not even one of my masters or PhD students worked with me without fellowship. I am very proud of that fact, proud of my institute, where incidentally I created 2 additional campuses, generated 1,800 crore rupees of funding. So that is my contribution. But I must be humble before you. Why I gave this general lecture? To make you aware of that what is this net zero. So if you go home and tell your mother what is net zero, I have achieved the purpose of giving this lecture. Thank you very much.
Thank you very much, Professor Yadav, for your mesmerizing talk. And I must compliment Professor Ranjana for having him here. And I heard him first time, I know him for long, but I know that he's a wonderful speaker and chemical engineer. And for the students, this is the new dimension what he has given to you. And thank you very much.
Society, we will have, and also I realize not many IITians are there, which is not a good thing. They must be there in that society. So I will need your help. We will celebrate in every, college. I have said that every Saturday afternoon we can have webinars, invite some exciting lecturers, talk on a theme and develop it. And for students, undergraduate student only for the entire batch 500 rupees, masters also 500 rupees, PhD is also 500, that does not mean prolong your PhD. Okay, finish it in time and join us. I'm telling you, college professors, college teachers, everybody should join Indian chemistry. This is 100 years, Acharya P.C. Ray, the first chemist, first entrepreneur because he started the Bengal Chemicals and Pharmaceuticals. Startup company, you must have heard about hydrochloroquine. Not only that, he is the one who worked about ionic liquids. Nobody knows about it. He published that paper. He was first person to work on ionic liquids. So remember that India has a great legacy and you should be part of this legacy. Thank you very much.
May I request Rawat sir to felicitate Professor Yadav sir with a memento as a token of respect and gratitude.
Speaker is Professor Shastri. Professor Shastri is the director of CSR NIST Jorhat. Before that, he was a scientist at IST Hyderabad, and before that he was a faculty member at Pondicherry University, and he has over 11,700 citations with H index of 53. He is the recipient of many awards and honors, including Shanti Swarup Bhatnagar Prize in Chemical Sciences in 2011, National Bioscience Award of DPT 2009, Swarna Jayanti Fellowship 2005 from PhD, PM Pitla Award 2001, BC Dev Memorial Award 2009. And I request Professor Sastry to deliver his talk. When 5 minutes are remaining, I will go on the dais. When time is up,
Hello, good evening. Still you are energized or our things? I don't think that I want to use this presentation. So now, what excites us? What is the thing that you have learned when you come to Nagpur? You need to have a take-home lesson, right? Because there are PhD students, there are MSc students, there are scientists who have accomplished, and we have a fantastic talk by Professor Ranjana about how we can look at the molecules and then play with the molecules and then elicit their biological response in a very systematic way, followed by Dr. Rawat, who is able to take a molecule almost to the clinical trials and very successful in 2-3 areas. Then we have very young and energetic G.D. Yadav setting stage on fire with energy and all these activities. So, what all we have to do is, when you come here to attend this conference, you need to have a take-home message. We need to understand that a country of 1,300 million people, where are we now in terms of original ideas? Where are we now in terms of our basic strength in chemistry? Actually, in one of the introductory slides, Dr. Rawat said around 1900, our average life expectancy is in 20s. So then, there was no need for us to work on neurodegenerative diseases like Parkinson or Alzheimer's or even heart attack, because there the most important thing is the hunger, roti kapda aur bukhar. Right? Now we have the sustainable developmental goals, 17 of them. Why do we have to worry about sustainability? Because we have already come to a stage which is good enough. Whether we sustain or not, that's a question that we have. Because about 50-60 years before, we did not talk about sustainability. Now, we talk about sustainability. And I am sure that if you are not very careful, you have to talk about survival. About 150 years before, if there is a power cut, will anybody be affected? But Dr. Ranjana, no, we don't even have power. When there is a power cut, there is no problem at all. They are not worried. Now, if you look at countries like Pakistan, already have a huge problem of energy. What is happening to the sustainability from the energy sector in India? It has become a joke in Europe that in winters they would like to have the room temperature with air conditioners around 25 degrees. And in summer, the room temperature they wanted to have it around 90 degrees. There is a single rule in some of the places where if you adopt a lifestyle where whether it is winter or summer, you will have a reasonable limit, at 24 degrees or 25 degrees. You will already save lots of energy. I would like to tell you that if the entire world adopts the lifestyle that India has, we need about 50% of the resources that are available. In contrast, if you are adopting the lifestyle of a United States, we need 3 to 4 times the resources that we have now. So the question is, how do you define development? The question is, how do you have a sustainable or a recycling economy which will make us happy from a different perspective? So, whether it is agriculture, whether it is health, whether it is energy, I completely agree with Professor G.D. Yadav that chemistry is a central science. It's absolutely important to realize that whatever we feel, whatever you put your hands on, everything is a chemical. Actually, the announcement that he has made, this was made by Royal Society of Chemistry. It was made to public that tell anything which is not chemical, we will give 100,000 pounds. Whatever you have, people say that is it chemical or not chemical. That's not a question to be asked. We need to ask the right questions. And the most important thing is, how is our education that is going to transform? If you look at the definition of health in the WHO, currently the definition is health is a complete physical, mental and social well-being, and not merely the absence of disease or infirmity. They are adding 2 more dimensions to it. Health is a complete physical, mental and social, emotional and spiritual well-being. There is a lot of work that we wanted to carry out to understand the disease pathophysiology and the way in which the relationships are going on between communicable and non-communicable diseases, which has to do with the nutrition and nutraceuticals, immunity and inflammation. There seems to be a lot of new chemistry and biology which is coming to the fore. And in this particular era, we have to see what are the opportunities that we have. And I feel that if some of the people are there, Industry 4.0 and Industry 5.0, we will have to understand how things are going. And most importantly, we have to realize the problem that we have with our education system.
I think the sectional president has given me the most difficult task to do afternoon actually. First, to chair the session of Professor G.D. Yadav, then good friend Dr. Shastri. Now, pivoted because we used to share the same floor in chemistry department of Delhi University. She is my colleague. But somehow she didn't like and she moved to GNU. So she is in GNU now, can't meet at Delhi. So after maybe 3-4 months we are meeting here now at Nagpur. So let me introduce Viva to the audience. Let me tell you one thing. She is one of the most hard worker I have ever seen. Delhi University and GNU की जो distance है 30 किलोमीटर है. And Delhi में 30 किलोमीटर का 1.5 घंटा 2 घंटा लगता है. She used to come to the Department of Chemistry, Delhi University before lunch, and after lunch she used to go to GNU to perform her duties there. I don't know how many of the students will be able to do that on a continuous basis. Okay, this is what she is. So as I said, she is with Jawaharlal Nehru University right now. Previously, she was faculty at Department of Chemistry, University of Delhi. Before that, she was with Ambedkar Center for a long time. And she has done postdoctoral work from IIT Kanpur. Total citation is about 4,700 with H index of 32. Guided 24 PhDs, and she has been honored with different awards and honors. She got 10th Darshan Ranganathan Memorial Lecture Award in 2018. No, students. Do you know who is Darshan Ranganathan? Nobody knows. So at least Google it and just read about her contribution. Okay. She got T. Nasipuri Memorial Award 2018, Mid-Career Award 2018-2021. With this, I request Diva to deliver a talk. When you have a 5 minutes before left, I will come, and when your time is up, I will stand.
Very good evening, and I know the session is late, and first of all, on the onset of my talk, I would like to thank Professor Ranjana Agrawal. Because of her, I'm here today, and so I should say, but she was kind enough to call me, and as well as I can see that my teacher, although he has never taught me, but I was mentored in the same department in Allahabad University, so Professor Tiwari is sitting here, and say some very senior Professor S.P. Singh is sitting here, and then obviously thanks a lot to Professor Rawat for giving such a kind, you can say such a kind words what he said, and so with all these things, and I would say that I would like to present my They don't want me. It's chemistry for sustainability. Should I go there? That's why he didn't present his work. That's why you didn't present his work. Your work. Okay, so fine. Generally, I was trained as a chemist, and then I became a biochemist, and I don't know when I became a medicinal chemist, and then a biologist. So it's just like that, and so it is on the basis of that. I would just like to thank. Start that I thought that I will tell you people today some of my journeys in the say different breakup. So the first, the very latest results which I wanted to present today, which I cannot present without the presentation, because it is not Professor Sastry. I'm not Professor Sastry, and I cannot be extempore. So basically, I must tell you that we have synthesized very recently certain C1- nucleotides and nucleosides, which are actually remdesivir analog. And I purposely included those slides today because I just wanted to address certain younger students because all of you are know very well remdesivir. So I just wanted to tell you a few things about the remdesivir and the development of the remdesivir also. So on the basis of that, what we did that we have recently synthesized few analogs of remdesivir. We have synthesized few analogs of remdesivir, and there is a phenomena in remdesivir, because remdesivir is actually a prodrug. So how many of you know prodrug? I don't know. Prodrug is a drug, is a form of the drug which is inactive form of the drug, and when it enters inside the body, it actually becomes an active drug. So conversion of a prodrug to drug is the phenomena which is followed in the case of the remdesivir. And I know. You know, I used to use your slides. You don't know that. I have put final now. यहाँ नहीं है. Final now. Amazed to know that while I started working on these remdesivir analogs, there were no starting material available in India. And say Professor Sastry belonged to earlier, his place of work was ISCT. So we got it from ISCT, lot of starting material. And we did so this says potency of a small molecule comfort critical human diseases a way forward. So I tell you, basically I work on medicinal chemistry, I work on against the different diseases, and I wanted to show some of the magic of the small molecules today. And so on the basis of that, so I'm not going into the introductory part of it. Is it? Okay. So in fact, my lab runs on the this benzimidazole generally, and you can say this is the bread and butter of my lab. And so we have been doing a lot of things with one class of molecule, and you will see that how one class of molecule is actually active against the different kind of can be used against the two different diseases. So in general, what I do that I do some kind of a targeted therapy and another kind of a non-targeted therapy. So what does it mean? That sometimes what we do that we synthesize a molecule against a target, and sometimes we randomly synthesize. For the benefit of students, I would like to tell you that most of the drugs which exist today are actually non-targeted ones. But today they say that one should do targeted therapy because of the several reasons, and because of the persons like Professor Shastri and a lot of other people who have generated a lot of software, a lot of data for us. We can utilize those data, and using that data we can synthesize a molecule much more easily as compared to the earlier. So the first thing which I just wanted to share with you, which I just introduced to you, that we have recently synthesized 1,1-disubstituted C nucleosides or nucleotides. What does it mean? That we have synthesized the remdesivir analog, and that was remdesivir act on the RDRP. What is RDRP? RDRP is the one of the enzyme in the in COVID, and it inhibits, and that particular enzyme is responsible for the replication of the virus. So as you can see here, that this is the structure of the remdesivir, and remdesivir was actually a dream project of several companies, and all those companies actually have developed this molecule, and they actually developed it against the Ebola. And but as soon as the COVID came, they started utilizing it. But what happened? They have done lot of modification, but they purposely never touched this pocket, which is called as the CNNN. So what we thought that why did not they touch it, and we did lot of docking, molecular dynamic simulations, and after that we decided that we will modify this particular portion of the molecule, and we will play with this molecule, and we will see, we will play with this functional group and see what happens. This was the portion which was going and binding to the backbone of the RDRP, which is an enzyme, reverse DNA, reverse polymerase. So what was, so I'm not going into the mechanism that how remdesivir actually works, but you can only say that the remdesivir causes a delayed chain termination of RNA, stalling the translocation. So what happened that whenever there is a replication, after there is a replication fork is synthesized, and then before that replication fork there are two RNAs are there, and that, so that is a complete process, and that happens in the mammalian cells also, and it happens in the virus also. So it actually stalls that process of the formation of the RNA. So, but the most important thing that we need to understand is that still people are not clear about the remdesivir mechanism even today. So then we did lot of work on this particular molecule, and we thought that we will make the modification at this place. And the modifications which we planned the heterocyclic rings over there. And so when we did the changes of this molecule, and please understand that this molecule has too many chiral centers. And the problem which with the remdesivir is that if you, what Gilead has never reported, but if you go into their patents, you will find it over, that when they used remdesivir alone, its half-life was very small, and when there was so that is why they always did the phosphoamidation, and they used actually a phosphate salt. So with all this thing, we started working on this, and we introduced a number of 6-membered and 5-membered ring at this position, and it was a very, very difficult synthesis. And there are two different diastereomer of remdesivir are known, that is the RP and SP. They call it RP and SP depending on the arrangement around the phosphate. Phosphorus group. So we have also synthesized a series of molecules. Before synthesizing those molecules, we have done all molecular simulation studies, molecular studies, and when we did those studies, we found certain molecules, certain rings which were giving us better score in the docking and the simulation. The complex was quite stable, so we thought that we will first try with those molecules. So after a long synthetic route, we have synthesized. I'm not going into the synthesis of this, but because it was a chemistry talk, so I thought that chemistry should be included over here. So this was we synthesized the one half, and then another half was also synthesized separately, and both the halves were then brought together, and then we got the final compound. So when we made this final compound, and what we did, that we have collaborated with we did some work in our lab, and then we collaborated with the Scripps Institute, with the Scripps Institute, and they have done a lot of studies with this molecule. The results were not great, but results were not bad also. But we have found that it was actually inhibiting the virus at 8 micromolar. So we have done the in vitro studies also with the enzyme, but then we did the live virus studies also. So I'm showing you the live virus results only over here. And the good thing with this molecule was that the toxicity of this molecule was lesser than the remdesivir. So in that way, so there are 3 things happened when we did this work. You can say that we could develop a project of synthesis of remdesivir in India, and we have used our indigenous substrates which were produced in ICT Hyderabad. We could produce certain intermediates which we can tomorrow give to other researchers in this area, and we could train the manpower also. So academics is something which not only runs around that what is your success rate, but it also most important specifically in the industries and the academic in the universities and academic institutions, that how much is actually your student has learned whatever you have done. The second very old project in my lab is all about from very beginning, from last 10 years, I have been working on the development of certain radioprotectors. And what is this radioprotector is? Radioprotector is a molecule which actually protects the cells against the radiation while you are giving the cancer. Giving the radiotherapy to the cancer patient. Why do you require such things? You require such things because when you are irradiating a tumor, the cells which are surrounding those tumor also get affected, and which is called as the radiation effect, and that is a very long-term radiation effects occur, and there are a lot of problems starts in the person, and you must have seen that such kind of people. So then what happened that patient develops a radiation syndrome, and when they develop a radiation syndrome, they are no longer, they cannot be treated, you cannot even give them chemotherapy. So every radiation oncologist is always looking for the radio protection, and this is a one area which is not very common, famous in India, but it is a big market and a big, you can see here that this is a 2.2 billion market today. So we have developed a molecule, it is almost 10 years, and as you can see that this is a big molecule. But while we brought it, we got patent, everything, we got it, we are collaborating with number of industries, as I have shown you, for the, getting the GMP data also. We are collaborating with one of the hospital also, so that we can take it to the clinical trial. But what I am today presenting to you is that while I was working with it, that I was forced by the BIREC and DBT to evaluate against the head and neck cancer. I did not want to do it, I was doing it against the gastric cancer. And I was forced because maybe a lot of you may not know, the students may not know that today head and neck cancer is the biggest problem of the India. And why this problem is there? Because of the candy consumption, because of the tobacco consumption. And we have been treating our patients with the modalities which are available in the West, and Western patients do not develop head and neck cancer or oral cancer because of the candy. They develop because of the smoking or alcohol or maybe some of the genetic issues. So all oncologists or radiation oncologists say that we treat them, we operate them, we do surgery, but what happened that they 50% of them either die, or 50% who survive, they just have to carry a saliva bottle along with them, and their life is literally miserable because we are not probably treating them properly. We are treating them with the Western regime, which is a wrong regime. So I was told that because you have a molecule, so you assign that molecule, you see that if it can work against the head and neck cancer. So then I'm not going into the detail. So what we did that we started using this molecule, and there is a problem in the head and neck cancer occurs that you have to give lot of radiotherapy. Surgery and radiotherapy, chemotherapy is less as compared to the other cancer. So when you irradiate the patient, what happened that the patient developed a kind of a symptom, and which is called as the xerostomia. Xerostomia means their salivary glands die and they are no longer able to develop, secrete the saliva, and if you cannot secrete the saliva, you cannot survive, you cannot eat. And so that is the major problem. So what happened that we evaluated this molecule, we did lot of experiments there, but basically one major experiment which I just want to share with you is that we have tried to what we did, that we have taken the patient tumor and developed a single cell from that patient tumor, and we injected it into the mice, and we have developed a patient-derived xenograft out of it. It's nothing a new thing, but it is difficult to develop it, and in India there was no patient-derived xenograft of head and neck cancer was available. Actrec was trying to do it, but they are still working on it. They don't have still ready. So we developed a patient-derived xenograft, and then when we have taken the patient-derived xenograft, and those mice, when we pre-treated with the DMA, and when we have we did not do any treatment, then you can see that if you look at this, this is this patient-derived xenograft which we have developed. This is nothing else. Here I'm trying to show you that there was a group of animal in which the mice was developed, and what you do actually when you put the tissue of mammalian tissue in the mice, you have to check that mammalian tissues is still carrying the mammalian characteristic. It has not been developed into the mice characteristics. So these are the things which I'm just trying to show you. But when we did these things and we tried to see the average saliva volume on this particular, measure the average saliva volume, so please see it carefully. I have just put some final results over there, that there is a control where the irradiation has been given. No irradiation was given, so this was the saliva volume here. When you are given 4 gray of radiation till 5 days, you can see that the saliva volume has gone down. But when you are given the DMA 50 mg per kg to the mice, as per the body weight, then you can see that the saliva secretion has increased. So slowly, when you increase the dose of the DMA, or you give the repeated dose of the DMA, the saliva secretion has increased considerably, as you can see. You can find it over there. So then we could find out that maybe there is some reason that it is actually acting on the salivary glands also, or it is increasing the salivary secretion. So then what we did that, so this is what you can see here, that we have done another experiment which is called as the tumor volume. So if you try to understand it, you can see that in the control, the tumor volume is quite high. In the pre-treated one, DMA pre-treated one, the tumor volume has gone down. And there is a Kaplan-Meier survival graph which says you that the DMA-treated mice have survived longer as compared to the untreated one. So this is one of the thing, and I will show you here, purposely this portion I have included over here, that you saw the structure of the molecule. I think you remember it here. So it is here, it is patented, you can find it out anywhere. So when you go into the, now I'll show you, change my gear, the gear of my work, and I will bring you from cancer to the antibiotic development. So when I bring you to antibiotic development, we have developed an analog of this particular molecule, and I'm trying to show you here that how can you inhibit a bacteria or kill a bacteria by inhibiting the bacterial topoisomerase. So because topoisomerase is again a enzyme which is responsible for the replication of the bacteria. Again, we are collaborating. There is a big group over here, and we have Anthem Biosciences, which is working along with. This is a CRO in Bangalore. So I'm not going into the antibiotic problem, but I just want to bring something to your notice, that whenever there is a WHO report which says that India has already entered in the say alarming stage, and by 2050 there may be say 30 million deaths may occur because of the antibiotic resistance. But what they have written there, which is very important and where few of our chemist front can only play the role, they say that whenever we try to develop the antibiotic, we take the original molecule and we try to develop a say analog of it. So what happens when you do the analoguing of the existing antibiotic, there is a mapping has been done by the bacteria of that molecule. So it is able to create the resistance faster as compared to the new molecule. So they say, please don't work, please don't try to develop any more say synthetic analogs of the existing antibiotic. Rather look for a new antibiotic or a new structure which is still not being recognized by the bacteria. So there are 3-4 recommendations over there, which anybody can read it on the WHO. So so on the basis of those things, what we did that we have filed we have gone to the DBT Biorec, and we have shown the activity of one of our molecule on some of the WHO priority pathogen. What is the priority pathogen? Priority pathogens have been launched by the DBT also, by internationally also, against which there is no cure. There are the latest drug, vancomycin, the 4th generation of antibiotic is also failing, and even the intravenous administration of the antibiotic is also not working. So we were lucky that they launched around 11 strains, and against 8 strains we could find some of our molecules which were acting. So we thought that okay, we should develop this molecule. And this is the topoisomerase structure. This is bacterial topoisomerase and the human topoisomerase. So for the, how much time? Okay, I'll finish before. So then what will happen that when you have the E. coli topoisomerase, there is so what is the concept here which I just want to put in your mind? That human topoisomerase is also responsible for say replication of the mammalian cell, and the bacterial topoisomerase is responsible for the replication of the bacterial topoisomerase. And topoisomerase is a kind of a protein which is actually so there are kind of a different kind of a bacterial topoisomerase are available, and when you inhibit one topoisomerase, so topoisomerase 1 then topoisomerase 2 will their say expression will increase, and bacteria is very smart and they try to manage these things by increasing the expression among them. Fine. But that can be handled. But the problem is this, that if you create a molecule and that also goes and binds to the human topoisomerase, then what will happen? Automatically the toxicity of your molecule will increase. So what challenge we had actually, that if we create a molecule that should show minimum binding with the human topoisomerase than E. coli topoisomerase. You may say that the structure of the E. coli topoisomerase and the human topoisomerase is different. Why are you worried? They are different, but their active site is not completely different. So it is not that you have an acidic triad over there also, you have some of the similar amino acids there. So the basic challenge which was there, so we synthesized around 150 molecule, and then we finally reached, and I tell you, this was a project which was done, I started in Ambedkar Center in 2004, and still we are working with it, and still we don't have the answer of several questions. So with all these things, I will just come to this straightforward, the data of one molecule, and you see that this is one of the similar molecule which I showed you earlier. So I just want to tell you that that the concept of the repurposing is true, that is fine, but whether you repurpose or you don't repurpose, but there is a misconcept in the chemistry that if you have a molecule, it goes and binds to somewhere, it cannot be, it will be really not a selective molecule, it is going and binding to everywhere, so you don't have to use it for another disease. But you can use it, but you have to make certain changes among them. So what we did, that we have collaborated with the sub-region hospital, and we have evaluated this particular molecule against the 600 strains and there, and you can see E. coli, S. aureus, and to our estimation, we were thinking that this will work better to gram-negative strain, but actually it works better against the gram-positive, and it has shown very good activity against the S. aureus, MRSA or the VRSA, that is the vancomycin-resistant strains and methicillin resistant strain. So we have done this molecule, we did MIC, minimum inhibitory concentration, and after doing this, this is a typical biochemical assay, and I think picture is clear but smaller in the size. But whenever you do this is called as the target specific design. So my target is assigned here, and I am trying to show you that it is inhibiting the activity of the topoisomerase. So what is only one picture I'll show you, that there is a supercoiled plasmid here. You have added topoisomerase, it has got relaxed, the DNA got relaxed, it has moved up. But when you have added the PPEF, it is not allowing the topoisomerase to act, and you are getting more supercoiled form rather than the relaxed form. So this was the whole crux of the work on which we have developed it. So there is, I collaborated with one gentleman, Professor Naresh Patwari from IIT Bombay, and he has done some kind of a, molecular simulations, which was quite special. But why I am showing this result over there, that by doing his molecular simulation studies, these are actually movies. I don't know whether it will work or not. So the only thing which I really want to show you. So what he did actually, that what I'm trying to show you, that when my molecule of interest goes and binds to over there, what it does, that it does not allow topoisomerase to open up and let the DNA enter. What is its work? What is its function? The function of topoisomerase is to relax the DNA. So what they have shown, that if you do a competitive assay with the PPF and the and the DNA, you will find that if PPF goes and binds there, it closes the gate of the, there is a gate formation occurs over there, this portion goes separate and this goes separate. And then what happens, that the DNA enters in the topoisomerase, and that is how topoisomerase relaxes the DNA. So what they have shown, that if PPF binds over there, it doesn't allow the entry of the DNA, and that is how it actually doesn't allow the DNA to replicate or to relax. So this is a model. Why it is important, that there is no model available. So now a person can use this particular model to if there is a chemist and they want to see that whether this molecule will work as a topo inhibitor or not, we can very easily bring your you can bring your molecule and see it, that whether it is inhibiting the gate opening of the topoisomerase or not. So as it happens that if you do anything on drug development, you have to bring it to the preclinical level. So what we did, that in our laboratory we developed the septicemia model and the thigh infection model of, and we have shown that, and we compared it with the levofloxacin, and this was the multi-drug resistant S. aureus. And we have shown here that if you don't give any treatment, that the infection, when there is no treatment, that all the animals die in 2 days. But when you give the pre-treatment, you can see here that the animals are surviving over there. Although the data is not matching with the levofloxacin, but it is still a very good data. And the best part of this molecule is there, that it shows very good activity against the multi-drug resistant pathogen as compared to the lab strain. So with all these things, I would like to I have already explained. So I just wanted to summarize that we could develop some molecule which acts against the radiation therapy, which acts against the bacteria. They are not same, but they are similar. And I have shown you some data on the remdesivir analog. This is the place where I work. But both the last two projects I developed in Delhi University, so I never forget Delhi University. And these are few of my say collaborators and my students. I'll show in the next slide. I have been generously funded, and then these are the few of my current students. With all these things, thanks a lot. Thanks.
For your thoughtful lecture. I must appreciate the audience actually. Right? It's 6 o'clock now. Hall is almost full. I must appreciate that. We are skipping the question answer session because of paucity of time. With this, I think we'll move to next lecture.
May I request Rawat sir to offer memento to Vibha ma'am for delivering such a beautiful lecture. Thank you ma'am, thank you sir.
The last talk of the session is the award talk, and time is given 10 minutes. It is Dr. Deepika Gig. Yeah. Deepika.
Good evening everyone, I am Ms. Deepika Givkar from School of Chemical Sciences, Devi Halde Vishwadhyalaya, Indore. Today I am going to present my research work entitled Cobalt Ferrite Magnetic Nanoparticles supported on hydromagnetite sheet embraced A3 coupling strategy targeted to synthesize fused benzimidazole thiazole skateboards. These are the contents which I am going to cover during my presentation, starting from general introduction to fused benzimidazole thiazole, Aim of the work which I have done, followed by the preparation and characterization of catalyst, reaction optimization, synthetic strategy, mechanism and its comparison with some reported methods, green evaluation, in silico studies and their validation through theoretical rationalization, spectral analysis of synthesized compound, merits of our work, and finally the conclusion. Fused benzimidazole thiazole is an important hydrocyclic scaffold isolated either from the natural products or accessed by synthesis. Imidazole ring is a constant of several important natural products like purine, histidine, histamine, alkaloid, and nucleic acid. It is result of the fusion of aromatic compound like fused benzthiazole to the 4th and 5th position of the pyrrole ring. As fusion with pyrrole ring enhances the delocalization of electron, which further enhances its property to a greater extent. Fused benzimidazole thiazole and its derivative find their multiple uses in medicinal chemistry due to their broad spectrum pharmacological activities such as anti-tumor, anti-cancer, antimicrobial, etc. This slide shows some commonly used drugs like levamisole, quizartinib has been shown which are used as anti-rheumatic, anti-cancer and PT analysis of Alzheimer's disease. In addition to drugs, these are also used as organic light-emitting diode as herbicides and optical patterning agents in detergents. The objectives of our work are to design and develop green strategy for the synthesis of fused benzimidazole thiazole derivative from easily accessible economical reagents to evaluate their biological potential and their validation through DFT studies. Firstly, we have synthesized the hydromagnesite sheet crowned with cobalt ferrite magnetic nanoparticles using chemical group precipitation method. In this method, firstly we synthesize the hydromagnesite sheet by the vigorous stirring of sodium carbonate and magnesium nitrate solution at room temperature, and the obtained white precipitate of hydromagnesite sheet was again treated with aqueous solution of cobalt chloride, ferric chloride, and sodium hydroxide at 100 degrees Celsius for 1 hour. Afterward, the obtained black precipitate was filtered and washed with mixture of water and ethanol until the pH is 7 and dried under vacuum at 60 degrees Celsius for 24 hours. After preparation of the catalyst, we characterize it with number of techniques. This is the FESAM and HR time images of our synthesized catalyst, which shows the size less than 50 nanometer and having good surface area. The chemical purity of the sample was tested by EDX analysis, and the EDX spectra presented here ascertained the presence of cobalt, iron, oxygen, magnesium and carbon as the only element present in the composite. This slide shows the X-ray reflection pattern for the same, which help in calculating the crystalline size, which is found to be 12.30, 16.97 and 14.78 nanometer for the composite cobalt ferrite magnetic nanoparticles and hydromagnesite sheet respectively, and the presence of the peak at around 30 degree corresponding to hydromagnesite sheet and the peak at around 35 degree corresponding to cobalt ferrite magnetic nanoparticles on the diffraction pattern of composite, which indicates the successful formation of corresponding hybrid material. The individual FTIR pattern for cobalt ferrite and hydromagnesite sheet were also recorded for their comparison with the composite. As we can see from this spectra, the bands are attributed to the stretching and bending vibration of CH, CO and OH group, and the peak ranges from 723 to 534 cm inverse are indoors to the deformation vibration of FeOH group, and the band at around 588 cm inverse are corresponding to the intensive stretching vibration of metal at the tetrahedral site between iron and oxygen, and the band at around 493 cm inverse is assigned to the octahedral metal stretching between cobalt and oxygen. The Raman spectra is also carried out for their comparison with the composite, and the calculated intensity ratio for composite and cobalt ferrite magnetic nanoparticles are 0.7965 and 0.7943 respectively. As we can see that it increases after doping of hydromagnesite sheet with cobalt ferrite magnetic nanoparticle as expected. The magnetic hysteresis curve were also recorded at room temperature for the cobalt ferrite and composite. At room temperature, as shown from figure, both the sample exhibit ferromagnetic behavior at room temperature, and the trends of magnetic saturation, retentivity and coercivity are seen from this table. The hysteresis curve reveals an increment in the coercivity value for the composite as compared to the bare cobalt ferrite magnetic nanoparticles. This increment may be due to the decrease in particle size with less magnetization. This slide shows the thermal decomposition profile of synthesized catalyst. We show the catalyst was entirely decomposed in two stages of the weight loss process in a temperature range between 25 to 1000 degrees Celsius, and the total weight loss over the composite in both the states is summed up to 54.50%. In order to characterize and determine the porous structure and surface area of the catalyst, nitrogen PET adsorption disruption studies were performed. As we can see from this graph, the specific surface area of the catalyst is found to be 75 meter square per gram, and the sample exhibit mesoporosity with a pore volume of around 0.4207 cm cube per gram, and the pore diameter with maximum center at 10.50 nanometer. keeping the economical and environmental issues under consideration, which are the usability of the catalyst and solvent for this protocol. And it could be seen from the graph that the recovered catalyst and solvent catalyze the reaction, an acceptable percentage yield, and yields of the product suggest no significant decrease in their catalytic activity and reaction efficiency. Thus, to synthesize this industry potent moiety, we need to monitor the reaction progress with different catalyst loading, optimize appropriate solvent, material and energy sources which minimize the cost and which also save our time and environment. We optimize the reaction using various protic and aprotic solvent at different time interval with varied amount of catalyst loading and found that we get excellent yield, that is 91% in PG400 as a solvent in the presence of 5 mole% of catalyst as shown in entry level. This is our synthetic scheme in which a mixture of 2-amino-benzyl-thiazole, patinate aryl benzaldehyde derivative, phenylacetylene derivative and a catalytic amount of cobalt ferrite hosted on hydromagnesite sheet in PG400 as a solvent at room temperature for the ambient time, that is 15 to 30 minutes, leads to the formation of our desired product. This two-amino reaction initiates with the condensation between 2-amino-benzyl and aryl benzaldehyde derivative followed by dehydration, which leads to the formation of shift space A. The A3 coupling strategy, where the basic sites of HMS promotes the formation of cobalt ferrite acetylide complex by the activation of proton from phenylacetylene. The A3 and the cobalt ferrite acetylide complex attacks on the iminium ion A to form intermediate B, and the latter undergoes 5-hexose decyclization instigated by an attack on the alkaline unit by benzthiazole nitrogen, which resulted in the formation of our fused product. This table shows the effect of varied substituent under similar fashion, yielding the corresponding antiseptic product in excellent yield, and it could be seen from the table that the electron withdrawing group resulted in better yield as compared to electron releasing group. To explore the a comparative account of literature protocol for the synthesis of fused benzimidazole-thiazole derivative amino present work are summarized in this table. As compared to the reported methods, our present strategy offers certain advantages such as simple procedure, easy workup, along with the usability of the catalyst and solvent. Today we all are facing health-related problem due to environmental pollution, but we can't simply blame to the automobiles and industry for polluting the environment. Rather, we, the researchers from laboratories, are found to be the major source for polluting the environment in the name and fame of research and progress. So it's our duty to incorporate such protocol which minimizes the pollution. In this regard, we have calculated the eco-scale parameter and found a score of 78, which is indicative of an excellent synthesis. We have also performed docking studies on our synthesized compound using Molecular Virtual Docker software. The docking score summarized in this table revealed that the compound 4E substituted with nitro group at the para position exhibit highest mole dock score among the series and standard drug ciprofloxacin. And it is fascinating to note that all the 10 synthesized derivatives display better binding affinity with the targeted protein as compared to the standard drug. The figure at the left is the dock structure of my synthesized compound, and at right is of ciprofloxacin, which is commonly used in the treatment of bacterial infections. This blinking arrow shows the antibiotic bind with cysteine and asparagine amino acid in our body, and our synthesized compound also bind with this amino acid, which shows our synthesized compound also mimic as a control drug. Theoretical tools have also been used to validate the stability and reactivity of the compound. For this, we have used Quotient 09 software and B3LYP 6311GDP basic sets. Basically, the stability and reactivity of the compound are appraised by global chemical reactivity parameter, comprises of total energy, chemical hardness, electrophilicity index, and electronic chemical potential. The calculated results summarized in this table reveal that the compound 4E with highest HOMO-LUMO energy gap are more stable among the series. We have also calculated the Fourier functions to rationalize and predict the reactivity profile of the molecule, which were evaluated on the basis of atomic charges obtained from electron density population analysis using this equation. Here, the highest positive value of Fourier functions indicate the most probable atomic site for electrophilic, nucleophilic, and radical attack. Characterization of synthesized compound with number of techniques. This slide shows the FTIA respect of synthesized compound inside big table along with their Okay, this is the 1H NMR spectra for the same. The merits of our protocols are synthesis of structurally diversified fused benzimidazole-thiazole derivative, recyclable heterogeneous catalyst and solvent, low catalyst loading, comparatively mild reaction condition, low E factor, high yield of the product, acceptable eco-scale value of 78, and atomic number that is 3.36%. Finally, I would like to conclude my work by saying that we have developed a simple, rapid and sustainable protocol for the synthesis of fused base emitters with thiazole derivative using efficient nanocatalyzed material and high efficiency, eco-friendly behavior and excellent reusability of the catalyst and obeying 3R concept put this protocol under the umbrella of green chemistry. Firstly, I would like to acknowledge my guide, Dr. Pratibha Sharma, Head School of Chemical Sciences, DAVP Indore, ISFA, RTM University for giving me this platform. All of you presented here, honorable professors, last but not the least, Almighty God. Some of my credentials, this is my list of publications. Thank you.
Thank you. Very good piece of work, good presentation, and she finished within given time. Any question from the audience or the experts want to ask? No. Yeah, please.
Size of nanoparticle, consequences such as what is it? And
Okay, I think.
There is enough different particles.
Okay, thank you. Yeah, thank you very much.
Thank you, Deepika ma'am. I now request Rawat sir to offer memento to Deepika ma'am. Thank you ma'am, thank you sir. Finally, moving towards the last part of the session. I feel blessed to be here. The world is full of diamonds and gems, and we are having some of them here today with us. I, Miss Priyanka, research scholar of this esteemed department. It was just awesome and honor to be in front of you all. Whenever you need me about anything, just don't hesitate, give me a call. I hope you have gained a lot of knowledge from this workshop and it will help you in making some decisions bigger or small. I tried my level best. Hope this communication will be with you whenever you recall. I now request our HOD, Karde sir, for giving a vote of thanks.
So good evening to all. As a head of the department, first of all, I consider it as a privilege to welcome all senior academicians and the resource persons and the participants coming over here. Actually, welcome should be done at the beginning, but we are feeling really very honored as well as motivated, and we received a feeling of blessings by interacting with such a very senior academicians. I have given a responsibility to express a vote of thanks. I have to say thank you to two chairpersons. The first one is Professor Rawat. We listened a very wonderful lecture of Professor Rawat. It motivated to many organic chemists, including myself. Second vote of thanks I have to give to Professor S.P. Singh, and I do not have any words to express vote of thanks about the Professor S.P. Singh. There are two major reasons. One is a personal reason, and second one is definitely the academic reason. I met Professor S.P. Singh in the year 2006, and he was an expert committee member of UGC MRP. And at that time, he has given me indeed a great encouragement to work on hypervalent iodine. And sir, I tell you, since last 20 years, I am carrying about 4 to 5 reactions on hypervalent iodine in my laboratory. So that is the thing I want to say. So thank you very much for that. And since there are many students, I must share two things about Professor S.P. Singh. We came across two wonderful books of organic chemistry. One is reaction mechanism, and recent one book is on pericyclic reactions. So I want that all the students should clap for such wonderful books written by Professor S.P. So thank you very much, sir, for coming. I thought in my life that one day I will meet to Professor S.P. Singh, and now time has this time that he is in our department. Thank you very much, sir. Thank you. Actually, the organizer has given me, first of all. Yes. So, I'm very sorry, sir, I forgot to express vote of thanks to Professor Tiwari, who chaired the first session, inaugural function. So, thank you very much, sir. Thank you.
Lastly, I thank you all for being with us. Thank you for blessing us with your presence, and I sincerely apologize for any inconvenience caused to you. Thank you so much. Have a great day.
There is one important agenda. Chemical sciences section election. So I will request Dr. R.D. Kaushik, former sectional president, to please conduct the elections. Thank you very much. And all those who are member of Indian Science Congress Association, life member before July, they will be the voters. So they please stay in the hall.
Over here who are either life member or simply annual member.